All right. Oh, it's working. Good. All right, ladies and gentlemen, good morning to a very exciting event. For those that don't know me, I'm Andrew Haleko. I'm the co-lead for the Devotion Network with John McGavick. And what we're presenting to you today is actually a fulfillment of something we pledged to do when we started, and I'm really delighted to see this roll out. And you'll hear from Cosetta here, Maliku, who's our trainee lead in Devotion. And so as you know, Devotion is a network of investigators, provincial investigators, interested in the science of the developmental origins of health and disease, with multiple pillars, including policy, clinical research, population health, and basic science, with the aim to integrate those. And I think we've made impacts and inroads in a number of research areas in the Children's Hospital Research Institute of Manitoba, CRIM. One of the things we pledged to do was develop a curriculum or a environment for training and inculcation, as it might be, in the art and science of DOHaD, Developmental Origins of Health and Disease. So conceptually, working with Cosetta and her group, we hatched the plan for a masterclass series, of which we now will roll out 4. Today is the first, with a wonderful speaker you'll hear from shortly. And the idea is to introduce us as a collective to the art and science, as I say, of developmental origins of health and disease research, and start building that culture from within and establishing it as a firm and sustainable platform here at the Rady Faculty of Health Sciences and, of course, within CRIM itself. This event gets organized by very many, and I sit through a couple of meetings, and then I get to stand up here with a microphone. But all the work really has been done by a number of people that you know. I'll acknowledge them now. Natalie Rodriguez, our executive director, here at the front, and I don't see Erica. Erica's here. I thought she'd run out of the room. Really have been responsible for coordinating this day, and Rachel's around getting signatures, and has coordinated the audio and video. And so I guess we're taping this for others to have access to, and maybe a little later Cosetta can fill you in as to where it will be, if it's going on our website or such. So welcome to this very exciting event. And as I say, this is the first of 4 masterclass series, which really are intended to introduce us to the different styles and approaches of research that are being used within the DOHaD domain. So we had a lot of discussion about appropriate first speakers and how do we launch this in the right way. And we fell upon a fairly strong consensus for our speaker today, which is Kristin Connor, who's you will hear from shortly. So Dr. Connor is an assistant professor at Carleton University, and she's in the in the Department of Health Sciences at Carleton, and she really is a DOHaD researcher, dyed in the wool, and I'm sure you'll be impressed with the breadth and the sort of scope of the work that she conducts. She has quite an impressive CV and list of accomplishments so far, including fellowship training at, boy, this is really small. Fellowship training in Auckland at the National Research Center for Growth and Development in Auckland, as well as the Lunenfeld Tannenbaum Research Institute at Mount Sinai in Toronto. So she comes from great stock in that regard. Her research looks at early life origins of disease and the interaction of environment with the host, as well as some genetics, but some the microbes and microbiome. I'm sure we'll hear a little bit about as well. So nutritional aspects of maternal health and fetal health are important, as well as infectious outcomes, which I gather is a new line of research for you. She is a fellow of the Society of Endocrinology and the International Society of Developmental Origins of Health and Disease, and she's also a Molly Toll prenatal research fellow, new investigator. So well-versed, and I understand, I haven't had the pleasure yet of actually seeing you in action, but I understand that she gives a wonderful talk that's interactive. So I think with that, I will hand it over to Kristin and welcome you to Winnipeg.
Thank you so much. I've been telling people that I've met that I never thought I'd have to come to Winnipeg to get nice hot weather, but it's been terrible in Ontario for the last 8 or 9 months, so thank you to whomever ordered this up for me. And I'd like to thank the organizers for inviting me to come and kick off the first of a few masterclass series that are going to be hosted by Devotion as you try to further shape the DOHaD community within Manitoba and build capacity for this type of research and training and knowledge mobilization. So I was given the task of giving you a bit of an overview about what DOHaD, or the Developmental Origins of Health and Disease, is, and with that I'll walk you through some of the key definitions in terms that are being used in the field, some which we may want to decide what they mean and how we want to convey their messages to those people that we work with and interact with. I'll highlight at a very high level some of the key epidemiologic evidence that really solidified this idea that the early life environment is critical for shaping our later life health and disease trajectories. And just touch on some of the mechanisms that may underlie DOHaD, but I know you've got a speaker that will be coming later in the series who actually will be talking a little bit more in depth about mechanisms. And then I was also asked to put out some questions or ideas about some of the emerging issues that might be happening in the field so that we can have a discussion on some of the big unanswered questions and perhaps start to think about the barriers to conducting DOHaD research. And then finally, I'll just build on a little bit of the great introduction and tell you about my DOHaD journey. So this isn't a research talk, and I would love to come back and give a research talk, so there's a hint for any of you who would like to invite me in the future, or you can come to Ottawa. But I know this is a bit of a diverse audience, and I think that this slide is important. It's one that I kind of lead off all of my talks with, and some of you may believe in this concept and understand this statement, but I still think it's important to say at the beginning of the talk here, and that's because I fundamentally believe that in order for us to know where we're going, we have to know from where we've come. And so we need to realize that the early life environment is critically important to our risk and resiliency across the life cycle. And we need to convince people, for those of us who believe in this, that this is important to everyone to understand and to apply to the work that they do. So as I said, I wanted to touch base on a few of the key terms and concepts that are thrown around in the field when we talk about longitudinal health and life course. And I think this is important to kind of put a grounding on because many of these definitions and terms are used in a variety of disciplines, from biology to medicine to psychology and sociology and even in economics. And these terms mean slightly different things to slightly different disciplines and slightly different people. And so I think one of the things that we need to do as a DOHaD community is really be clear in our choice of words that we use and how we want to convey the messages that these words might represent. So perhaps as you think of your own research on a longitudinal scale, and you think about growing this hub that you already have here in Manitoba with respect to DOHaD, you can explore these words and these terms and their definitions a little bit more, and then you can come back and tell the rest of us which ones we should be using and what they really should mean to those of us in the DOHaD field. So I'll just touch on a couple of these so you can see what some of the similarities and differences are across disciplines in their use and definition. And these definitions have been put out by scholars who know much more than I do. And it's important to know that even though these seem to be some common definitions, they're not necessarily agreed upon by everybody, even within a discipline. So the first is life course. We use that word a lot in DOHaD, and some fields define it as events marking transitions and trajectories of roles extending across the lifespan. And so this could include things like entering or leaving school, getting married, and having children. Another definition of life course, and the last two are probably more along the lines of what we traditionally think of in the DOHaD field. Another one is investigation of the changing environment and how it has developmental implications for that individual. And finally, one that seems to be clearly linked to DOHaD, definition of the life course is linking early life experiences to later life outcomes. But then the word life cycle is also used, and some people think, oh, well, what's the difference between life course and life cycle? Aren't they the same? Well, in the very traditional biological definition of it, life cycle actually means the continuous and irreversible sequence of changes undergone by an organism from one primary form to the development of that same form. But in sociology, it means something slightly different. It's a series of stages across the lifespan, such as childhood or middle age, that are socially constructed, and these characterize the existence of an individual. So again, slightly different approaches to what life cycle means. And we can't just think about someone's lifespan, so the period from which you're conceived until you die, but we also need to think about our life history when we're thinking about DOHaD. And life history means slightly different things, again, to different disciplines. Classically in biology, it's thought to be a series of living phenomenon exhibited by an organism in its course of development from inception to death. So it's got a very discrete window of when we're alive. In sociology, though, this takes on a slightly different meaning, where it's the lifetime chronology of events and activities that typically combine information from a variety of contexts, including one's family, residence, psychological well-being, physical and mental health. So again, you can see how these definitions are different, and in our field, considering there are many different disciplines that actually are working in DOHaD, even if they don't realize it, we need to get this terminology right. So I would suggest that maybe we use the term life cycle, but appreciate that life cycle goes beyond the definitions that I just provided to you, and that perhaps we need to extend our view of the life course beyond an individual's lifespan, so from when they're conceived or born until when they die, and think about future generations. And so the life cycle of an individual doesn't just end at their death, but perhaps goes forward on into subsequent generations. And I know some people in the audience and at the Institute here do transgenerational work, and it's very difficult work to do, so kudos to you on that. But I think this is a really important thing that we need to think about when we think about life cycle, thinking beyond the end of life of an individual. And I also think that we need to think about life cycle in the context of what happens before we're even conceived or even born. So this includes our life history. And so that means that we need to recognize that a healthy start to life leads to a healthy and prosperous adult life, and that healthy and prosperous adult life will lead to a healthy start to life in subsequent generations. Okay, so our life cycle, fundamentally, I believe, is more than just the period in which we're alive. It's our life history, it's our life course, but it does also include our lifespan. So this means that we need to be aware of our environments across this entire life course or life cycle. And this includes the preconception environment. This is a really critical period in our life that's not only important to mothers, but also to fathers, to adolescents, to boys and girls, long before they're contemplating a pregnancy. We need to consider the period very early around conception. And although this is a time when, say, the nutritional requirements of the developing embryo and early fetus are very small, it's still very important for maternal health and the adaptations that occur during pregnancy, as well as setting the stage for proper placental development, which we know has major implications for the development of the fetus. And then also in the fourth trimester, so the postpartum period. This is a period that doesn't get a lot of love in a lot of fields, but we have some experts who are really paving the way for looking at how critical this period of life is, not only to maternal health and interactions between the mom and the baby, but how it is a window that can program long-term health and disease trajectories. And I think this window might be even one of the most important, because the fourth trimester, that really early postnatal period, is a window that offers us the possibility to correct trajectories that have been set prenatally or even periconceptionally. And it also offers us the possibility to keep people on the right track if they are on a good trajectory very early on in life. So that optimal course can really be shifted in that fourth trimester or postpartum period. But how do we really know that events in early life play a role in later life health and disease? Well, these concepts about our life history and the environments that we're exposed to shaping our development and disease outcomes are not concepts of modern times. You might know of these guys up here. The one on the left, his name is Aristotle. He was a Greek philosopher and scientist, and he wrote many key pieces, one of which was called the Golden Mean. Maybe you remember this from elementary school. And the basic premise of the Golden Mean is to avoid the extremes. And this idea of avoiding the extremes is actually fundamental to DOHaD. We don't want to be born too soon or too late. We don't want to be too small or too big. We don't want to grow too quickly or too slowly. We don't want to have too few nutrients or be in an environment where there's excessive nutrition. Okay, so Aristotle was on to something, and we need to think back to life history in terms of philosophers when we think about what DOHaD means. And the guy on the left, Hippocrates, again another Greek, known as the father of Western medicine. And like Aristotle, he produced a lot of important teachings and pieces of work. And I think that he was actually probably the first to think about the developmental origins concept. He didn't call it that. He called it On the Nature of the Child. This was one of his really important pieces of writing. And he was probably one of the first to document that diseases were not caused by superstition or the gods, but they were actually a product of our environments, of the diets that we ate, and our living habits. So these concepts that we're really trying to understand, people were thinking about long time ago. And so then this brings me to probably a little bit more modern definition of developmental programming. And the classical definition of this that we use in the field is one where a stimulus or an insult operating at a critical or sensitive period of development could result in long-term structural and functional consequences for the organism. And many of these stimuli or insults fall under these kind of major themes. They could be exposure to xenobiotics or nutrients, inflammatory processes, and stress. And of course, whilst these are biological factors, they are all influenced and brought about by social and environmental contexts. But really to understand how we know early life environments shape our later life health trajectories, we have to go to the epidemiologic evidence. And some of you may know a few of the studies that I'll briefly talk about here, but this was the first piece of evidence that really showed what happens in our early life may play a role in our later health outcomes. And this study was the initial studies were based on key work from this woman. This is Ethel Margaret Burnside, and she was the Chief Health Visitor and Lady Inspector of Midwives in Hertfordshire County in the UK. And because of the meticulous work that she did with her fellow midwives, making a very meticulous record of all the babies born in Hertfordshire County in a particular period of time, and maintaining these records for so long, some scientists and clinicians in the UK were able to extract really important information that eventually led to what was called at the time the fetal origins of disease hypothesis. So the Hertfordshire records were a record that recorded the birth weight at 1 year, and illness and development of every baby born in Hertfordshire County between 1911 and 1948. And a group of scientists came across these records. David Barker, you may know him, he's one of the fathers of DOHaD, and he and his team recognized that the most common cause of infant death at the beginning of the 20th century was being born of low birth weight. And they hypothesized that if an infant was born of low birth weight, but survived infancy and childhood, as an adult, they still may have some sort of suboptimal or compromised health. And so what they did was they were able to identify individuals who were recorded in the Hertfordshire Records Ledger and track them into adulthood and following up morbidity and mortality records. And the seminal publication that first came out showed that babies who were born at the low end of the normal birth weight range. So these are not pathologically growth restricted babies. They're just at the low end of the normal birth weight range. They were at greater risk of dying from coronary heart disease as adults. And this relationship between being born of low birth weight, surviving infancy and childhood, and growing into adulthood and having adverse effects held true also for rates of type 2 diabetes and altered blood lipids. So this was really the first set of studies that showed the early life environment, and then in this case birth weight, which was a surrogate marker for something that didn't go well in the womb, could be linked to later life health outcomes. But it's not just about size at birth. It may also be about the trajectory of growth of the fetus in the womb or that infant in the early period after birth. And a lot of the Nordic countries, again, have these wonderful birth records, and we're learning a lot of information from them. And this is another example of that, where in Helsinki, there were meticulous birth records collected over a span of many years. And inspired by the work of Barker and colleagues, investigators went and looked at the Helsinki birth records and then tracked individuals from those birth records into adulthood. And this is just one example of some of the findings. So to orient you here, the horizontal line you see almost in the middle, that's the mean Z-score for the population. So if your data point falls below that level, in this case, you are shorter, lighter, and have a lower body mass index than the average. If it falls above that level, you are taller, you are fatter, you have a higher body mass index. And what was found initially from the Helsinki birth cohort was that boys and girls who would later get coronary heart disease as middle-aged men and women initially grew very poorly from birth to age 2, and then you can see on the far right a vertical line denoting the age 2 mark. And they also grew very quickly after age 2, particularly when BMI was measured. And ultimately, the relationship that seemed to be strongest in predicting coronary heart disease events was being thin at age 2 and having a high body mass index at 11 years of age. So clearly there's a relationship between size at birth and perhaps even growth trajectories in early life that can influence or be linked to later life health outcomes. But again, to reiterate, we're not necessarily talking about pathologic conditions here. We're talking about markers of something that happened in utero or in the early postnatal period that are operating under normal physiologic processes, which means when we think about mechanisms, we need to understand that we're learning about mechanisms that are also not pathological or not from abnormal pregnancies, but something that's going wrong in a normal process. And really, we know that it's not just about our size at birth or how well or poorly we grow, but it's this idea of what we experience as an individual very early in our life that can shape our later life health outcomes. And some of the most convincing evidence of this, again, going back to epidemiology, came from a very tragic natural experiment. Some of you may know about this. This is the Dutch hunger winter. And this was a period in 1944 to 1945 during the war. Because of German Nazi rule, there was an embargo on rail transport into the Netherlands at the same time as severe winter conditions. And so a population that had been normally well-nourished suddenly went into acute famine. Now, at the spring of 1945, Allied forces liberated the Netherlands, so rail transport could come back in, and this population became well-nourished again. And just like the work of Barker and others, Tessa Roseblum and other investigators in the Netherlands went to the meticulous birth records that were still recorded during this awful time in history during the war, and tried to see if they could find out what was happening to the individuals who were born from pregnancies that were manifesting during the Dutch hunger winter. And what they found was that the time during which a woman was pregnant and experienced the famine determined the spectrum of disease phenotypes in her offspring as adults. And ultimately, these findings showed that the experiencing the famine in the first trimester of your pregnancy happened to have the most adverse outcomes on offspring when they grew into adulthood. And this included everything from schizophrenia to depression, poor cognitive function, also metabolic derangements like atherogenic lipid profiles, increased rates of obesity in females but not in males, increased rates of type 2 diabetes, and now we're learning more that cancer incidence is different in these individuals who are exposed early in life. In contrast, women who experienced the famine in mid to late pregnancy, they had babies who were born lighter and shorter at birth. Okay, so the first group, actually the babies were born at term and of normal weight. But this probably doesn't surprise most of us who know about fetal development, considering most of fetal development is happening or growth is happening in the later part of pregnancy. So not surprising, these babies might be lighter and shorter when they're born. And you can see the drastic difference in terms of adverse health outcomes in these individuals as adults. So this established a role for nutrition as being something that's really powerful in shaping our development and health trajectories, but it also established something called critical windows. And the take-home message here is that probably the experience of that developing embryo and fetus very early in pregnancy is more important in terms of the antenatal critical window than other aspects of or other stages of development. Now, this is true potentially for nutrition and famine, but it may be very different in terms of when exposures happen when we're talking about stress exposures or something else. And there was another very unfortunate catastrophic event that many people don't know about that is now being looked at through a DOHaD lens, and it is the Great Chinese Famine. Now, this is actually one of the largest famines in human history. There were 20 to 30 million deaths, and compared to the Dutch hunger winter, the Great Chinese Famine was longer in duration. The population was generally not well nourished even before the start of the famine. Food availability was more severely curtailed across the nation, and there were different types of exposures ranging from very severe to more moderate famine. And what you can see here on the left is a Chinese population pyramid showing a massive decline in the number of people born between 1959 and 1961, so the period of the famine. And this is reflected in very high infant mortality and very low birth weights during this time. Now, there have been two nationally representative surveys, cross-sectional surveys that have been conducted in China since the famine, 40 and 50 years after the famine, in 2002 and 2010 to 2012. And as I said, researchers are now starting to look at these data from a DOHaD lens. And this is one paper that's just recently been published looking at a risk for central adiposity in this population. And there are almost 19,000 individuals that have been surveyed in these national surveys. And if you compare individuals who were exposed to the famine in the womb as a fetus with those who were exposed to the famine in infancy, with those who were not exposed to the famine at all, you see differences in risk for central adiposity. Particularly, there's an increased risk for having greater central adiposity if one was exposed as a fetus, is female, and interestingly, is now living in an urban area and has high education. And I say interestingly because I think this sets up the idea of another concept I won't really get into today called the mismatch hypothesis. And that concept really suggests that the degree of mismatch between that prenatal environment and the postnatal environment in which the individual is born into and lives in, the degree of that mismatch will determine the severity of the later phenotype. So perhaps these people who were exposed to this super awful event in utero or in early infancy, but then moved to urban areas, got a higher education, presumably then had more access to resources, food, perhaps even poor quality food, energy dense but nutrient poor. They may have the most severe mismatch that's occurred, and that may manifest in a greater risk for central adiposity. So you can see now how events like the Dutch hunger winter and the Great Chinese Famine revealed something that we like to call critical windows. And these are periods of development where there is phenotypic plasticity. We know in these critical windows, in part what defines them is interactions between the environment as well as the genome, the epigenome, and we're learning now more about the microbiome. But we also are now learning that the period that an individual responds to these external factors is not as discrete as once thought. Our responsiveness can actually extend across our life cycle. And what defines a critical window, importantly, is the dose and timing of that exposure. And that will then have an effect on the resultant phenotype, whether that's at the molecular level or the morphological level of our tissues or the general physiology of an individual. So dose and timing of exposure are important. And I think it's also important to state again here that timing just isn't about the prenatal or even the periconception period. But again, we have to think about the individual long before pregnancy is occurring. We need to think about that critical 4th trimester. And we also need to think about exposures not just being a single event that happened, where now we are exposed to many different things throughout our life, lots of stressors, lots of secondary challenges, and second hits and cumulative effects. So timing and dose, in terms of their definition that's used in critical windows, is now expanding that definition. And something that I probably will mention a couple of times, I think we really need to try to reframe our position in the DOHaD field and think about things more in a positive light. And that's also true for critical windows. Timing can also mean the timing of interventions as well, all right? There are critical periods during which an intervention may have greatest effect, and work from the Nobel Prize-winning economist James Heckman suggests that you get a better rate of return on investment, at least in terms of improving human capital, if you intervene in those very early years of life. And also, exposures aren't just negative ones. We need to think about exposures from the perspective of those that also establish and support resiliency in an individual or group. So let's not always think about pathology. I do it, I think we all do it in the field, but let's start to think about things more positively. So to summarize kind of this section, it really just means that our disease risk curve looks like this for most of us, where we, for especially non-communicable diseases, are at very low risk of having these for the most part of our life until much later into old age. But, you know, if something happens, like if we are exposed to an inappropriate stimulus, or even if we're exposed to an appropriate stimulus that occurs at an incorrect developmental window, this can actually shift our early phenotypes. It can alter the structure and function of our organs and tissues. It can mean that we're born too soon. We may be of a suboptimal size. We may grow too quickly, and we may have other impairments in our immunity and metabolism and our brain function. And these adaptations to these adverse exposures may initially be appropriate, because ultimately the fetus wants to be born and survive ex utero, all right? So they may be very short-term, appropriately timed adaptations. But in the long term, what this means is that the individual is programmed. And so this means that the disease risk curve shifts such that diseases now, particularly non-communicable diseases, are showing up earlier in one's life course and perhaps even to a greater effect. But how does this really occur? So we've got evidence linking early life environments to later life exposures. I told you that initially this might be a positive adaptation to allow survival, but long term it shifts our disease risk. But how? What are the mechanisms that are underlying these? Well, to answer this question, I think it's important to recognize that, especially when we are trying to uncover mechanisms, we are limited by what we know. Seems like an obvious statement, but sometimes we forget this. And I wanted to show this figure from some of the work that we're doing on a really massive and complex project that I won't have time to get into, but it tells us something interesting. And these are systematic reviews and meta-analyses that have been done in the last 10 years that analyze various exposure outcome relationships within a DOHaD or life cycle framework. And what we can see here is that there's a lot of scientific effort that has been put towards understanding the effect of nutrition in early life, and these could be maternal, paternal, breastfeeding, postnatal nutrition. There's also been a lot of effort put towards parental metabolic health, but much less so put towards things like environmental exposures, so exposure to toxicants or drugs, and much less so looking at psychological exposures and social determinants. So there's a real gap in our understanding of fundamental exposures and the context that could provide resiliency or risk for later outcomes in an individual. And this means that when we talk about DOHaD, the mechanisms underlying DOHaD, and when you think about these mechanisms even in your own work that you do, I think we really need to remember that we know what we know, but we don't know what we don't know. Okay? Makes sense? So what are some of the mechanisms that underlie developmental programming? Well, these are probably the most widely studied and kind of cemented based on the evidence that we have. It includes exposure to hormones, particularly stress hormones, nutrition, as we saw from the examples of the famine, but severe famine isn't the only way that one can be programmed nutritionally. We know epigenetics, genetics, and we're learning more about how our microbiome may be a key player in shaping these associations. We know inflammatory processes, oxidative stress, and the failure to repair DNA damage is also a critical mechanism for programming outcomes. And we're starting to learn that the pre- and periconception conditions and postnatal environments are also key players. And I'm going to give an example, or 3 examples actually, about some of these mechanisms. Again, knowing that someone is coming later to talk in detail about these. But before I do, I think it's important to point out, and I usually say this to people who don't know anything about DOHaD, or to students in my class who get really worried when they hear about, oh my God, did my mom eat that or not eat that, or I was born preterm. Just because you've been exposed to some sort of stressor, some sort of adversity, doesn't mean that you're going to grow poorly, become obese, have type 2 diabetes as an adult, or a variety of other outcomes. Fundamentally, part of what shapes and determines this later phenotype is based on interactions of the host, the environment, our genes, our epigenome, and obviously now we're learning about our microbiome. So in the context of time that we have today, I just wanted to highlight 3 different situations. One is a classical biological mechanism, one of the earliest and most well studied in DOHaD, and it's also a very powerful programming mechanism. The next is something that I think is really, really cool. It's not really studied very well, and certainly not in the DOHaD field. I hope it is studied more, and I actually think my good friend Megan might be the key person to look into this. But it's an example that I'm using of programming of behavior, because we need to remember that behavior is very important in shifting our health trajectories. And the last example is something that is fairly new and not well understood, not in DOHaD, but just generally. And I think it speaks to us being very adaptable and malleable in the field so that we can try to understand new and emerging issues that are coming out. And this example will also speak to mechanisms that might be operating very, very early in our lifetime that can set our later life health. So the first mechanism, the classical, most well-studied, powerful programming mechanism, is exposure to glucocorticoids or stress hormones. And why did people, not that long ago think that glucocorticoids were really important in developmental programming? Well, it came from some classic information and studies. We know that the hypothalamic pituitary adrenal axis, or the HPA axis, is altered. Its function is altered in overweight adults and those who have obesity. And the adrenal gland in the HPA axis is the key component that produces these glucocorticoids, particularly the stress hormone cortisol. That is a powerful programming stimulus both in normal growth and development, but also in suboptimal growth and development. We also learned that from rat studies, if you take an obese sucker rat and you remove the adrenal glands in this rat, you can actually ameliorate the obese phenotype. So something being produced from the adrenal gland that is no longer in the body is actually preventing the obese phenotype from persisting. And we also know that our organs are highly sensitive to the effects of glucocorticoids because they contain glucocorticoid receptors. So the ligand can bind to the receptor and exert its effects. And this is true also in the context of pregnancy and development. So the fetus has glucocorticoid receptors. They don't just appear when we're adults. And these glucocorticoid receptors and the interaction between the ligand and receptor in fetal development are very important in normal pregnancy because glucocorticoids help to mature the organ systems in the fetus. And they do this at very appropriate times during development of these various organs. And one of the most important organs that glucocorticoids help to mature are the lungs, so that the fetus can breathe and survive on its own ex utero. And the management or the regulation of exposure of the fetus to glucocorticoids in pregnancy occurs at the level of the placenta and the placental 11-beta HSD enzymes, so the 11-beta hydroxysteroid dehydrogenase enzymes. The most important in the placenta is 11-beta HSD2. It inactivates active cortisol into inactive cortisone. and therefore protects the fetus from high levels of glucocorticoids that circulate in the maternal compartment. Now, the placental glucocorticoid barrier naturally declines with advancing gestation. And this is something we want to happen because, again, we want these glucocorticoids to cross into the fetal compartment, target these organs, particularly the lungs, so the fetus can then have mature lungs, breathe on its own ex utero. But we and others have shown that if you undernourish a mother during pregnancy or in the periconception period, or if you find out that a mother has been dieting to lose weight before pregnancy, you can actually see a reduction in this protective placental glucocorticoid barrier. And this is associated with a reduction in the barrier at inappropriate times in pregnancy, which leads to higher levels of cortisol in the fetal circulation. It's associated with shorter gestation length in sheep, mice, and humans. And at least in some of the human and animal studies, we know that it's associated with altered stress responsiveness in childhood and adolescence. So we think that the way that undernutrition exerts its programming effects happens through the actions of glucocorticoids. Oh gosh. So we also know in humans that glucocorticoids are a powerful programming stimulus independent of nutrition. This was another very unfortunate natural experiment that happened right here in Canada. Some of you might be too young to know about it, but it was what's called in Canada the Quebec ice storm. But it was an ice storm that happened in 1998, and it affected Eastern Ontario and Quebec, as well as northeastern United States. And it was a very catastrophic event because there were millions of people who experienced the ice storm and then were without power and water and heat for up to 40 days in some areas. And it was considered a highly stressful event. Now, there's some scientists in Canada who really, I hate to use this term, capitalize on these natural experiments, but are doing very important work. And when the ice storm happened, they were able to recruit women, over 150 women and their infants, so these DOHaDs, and follow them through not only childhood, but now into adolescence and the teenage years. And amongst many other findings, what they showed was that the ice storm can actually have negative effects on health outcomes and development of the offspring, such that babies or mothers who experience the ice storm in their second trimester of pregnancy especially had children who had negative cognitive and language development from as early as age 2. Mothers who experienced the ice storm in their first 2 trimesters of pregnancy also had babies who were born earlier and were of low birth weight. And mothers who experienced the ice storm, and particularly those who indicated they had lots of stress, so the severity of stress was really high, they had children who had increased insulin secretion at age 13, and there was a dose response with this. Sorry, I don't know where my mouse is to go. Did it shift forward? There we go. Okay. And so these researchers have also looked at epigenetic regulation and changes in the offspring from mothers who experienced the ice storm. And what they found was that maternal objective stress, so this is stress like how many days were you without power or water or heat, as opposed to subjective stress, which is much harder to study. Objective stress was associated with DNA methylation changes in adolescence, and many of the pathways that were altered in differentially methylated genes happen to be related to pathways regulating immune and metabolic function. So again, you have a situation here where epigenetics is playing an important role. And I think epigenetics is a very powerful biomarker or signature that can help us to uncover programming mechanisms. And then as a result, epigenetics can help us to point to intervention or prevention strategies and may be useful as a target that way. The second mechanism that I want to talk about is related to this idea of programming of behavior. And there's been a lot of work that's looked at the central brain and how regulation of appetite, particularly at the molecular level in the brain, and energy expenditure can be associated with later rates of obesity and type 2 diabetes, particularly when this is looked at in animal models. But the hypothalamus and the brain are not the only areas of the body that help to contribute to our appetite regulation. We also need to consider how we develop our preferences for food and our flavor learning. And I think this is a really, really cool area to study. And like brain circuitry, our flavor learning pathways are laid down in utero. So the functional parts, like our taste receptors, they acquire the structural characteristics of an adult taste bud, and they're functionally mature as early as mid-gestation and certainly by the end of gestation. So it's not just the brain that's controlling appetite, but our taste centers are being programmed early in life. And actually we know that there's a mismatch in the world that we live in right now and the world that we evolved from in our life history. So we used to be living in a world where food was scarce, and we were always looking for that elusive calorie. And that usually meant finding things to eat that were salty and sweet, they were more calorically dense, and avoiding things that tasted bitter, because these were often plants and other components that would be very toxic to us, eventually kill us or make us very ill. And so evolutionarily speaking and thinking about fitness, we want to make sure that we're not dying or getting sick, but that we're actually living, that we're consuming what we need to be reproductively fit and carry forward and reproduce. And so that means that probably in our evolutionary past, we've been programmed to like salty and sweet things. Well, it also means that we've been programmed to dislike bitter things. What kinds of foods do those represent? Not liking your Brussels sprouts and liking high calorie dense, nutrient poor foods. So I think we need to keep in mind that there may be a life history that's embedded in our preferences for food that we may not be considering. And we also know that we learn about flavor by building on familiar flavors and repeated exposure to flavors. And that's how we develop our flavor preferences. And this occurs right from in the womb. So we know that the fetus swallows large amounts of amniotic fluid, particularly towards the end of pregnancy. We know that what the mom eats flavors that amniotic fluid. And as I told you previously, the taste receptors are laid down and mature in utero. So that means that the fetus can actually experience these flavors in utero as it swallows this amniotic fluid. We also know that what the mom eats flavors the breast milk, and flavors in breast milk can actually influence an infant's exposure to and liking of and acceptance of a particular food. So although we may not be able to change our embedded biology, there may be consequences if we're not being exposed to different types of foods, different varieties, different flavors of foods. And this could be potentially put into context of an infant who is formula fed and instead of breastfed. There'll be a break in that repeated exposure flavor learning chain. Or if the weaning foods that a child is experiencing are different from those that it experienced through breast milk or through the amniotic fluid. So again, I think this is really, really important because it's showing that there can be programming of behavior that may be linked to some of the classic disease outcomes, and particularly non-communicable disease outcomes that we experience. Sorry, I don't know what's going on, if there's a weird thing on your screen, but I apologize for that. The last mechanism that I want to touch on is this idea of what's actually happening very, very early in life, and us as a community of researchers and scientists and clinicians being very adaptable to emerging issues in our field. And that is, what is actually in that microenvironment that we are conceived in? This is mostly a black box, and this is really important, I think, because we are now facing a huge issue of subfertility and infertility all around the world, including in Canada. And although there are methods to combat this, very successful methods like assisted reproductive technologies, this may set up an artificial environment that is very different from that of natural conception. And you can see here a table showing some of the major differences between key factors that determine successful insemination, fertilization, and implantation, and how they're different in natural conception to assisted reproductive technologies. And these may include the types and levels of hormones that one's exposed to, how ovulation occurs, the actual environment that fertilization occurs in, so in utero versus in a cup or a dish. So the natural environment and the ART type of environment are very different. And although it's very difficult to separate the effects of assisted reproductive technologies from infertility itself, because a lot of couples who come in seeking infertility treatments also have other comorbid conditions. They might have increased parental age. They might have conditions such as obesity. So it's very difficult to really tease these apart, but the evidence is getting a little bit clearer. And what we're finding from early studies in animals as well as in humans is that the artificial environment has implications for embryo quality, altered fertilization and implantation and pregnancy rates and live birth rates. It seems to be associated, at least to some extent, in rates of low birth weight or small for gestational age. Whether this is a situation that's occurring just because it's the most well studied to date or it's the major programming outcome, but there appears to be a lot of issues with endothelial and vascular dysfunction in offspring who are conceived by ART. Some studies have started to look at teenagers, and they're showing those who conceive by ART are greater risk for poor body composition. They've got altered cortisol responses, and they may have different abilities to control their insulin and glycemic mechanisms. And why is this? Well, we don't yet know, but maybe it's because of the microbes. The environment that we are conceived in is not sterile. We used to think that this was a sterile environment. We're learning now it's not. There's studies that are now looking at the seminal plasma and finding that there are distinct microbial communities that exist in seminal plasma, and the composition of the microbes in the seminal plasma is associated with semen health and male fertility. Studies have also looked at communities of bacteria along the entire female reproductive tract and found that at different parts of the reproductive tract there are very distinct microbial communities. And specific taxa in each of these microbial communities in the female reproductive tract also associate with things like reproductive tract diseases, rates of implantation, rates of pregnancy and live birth. So clinically, the microbiome needs to be considered in fertility, and this is probably very true for situations like assisted reproductive technologies where fertilization is not happening in the normal microenvironment that it is in natural conception. So the microbiome, as many of us know, is really an emerging player in DOHaD. We're starting to learn a lot about it, but we still don't have a lot of information. And ultimately, it represents another genome that we need to consider in developmental programming. And this is because ultimately the microbiome that gets seeded in that infant will play a profound role in shaping its lifelong function in terms of immune system maturation and function, functioning of the metabolic system, and we're learning more now also about the connection between the gut and the brain and the neurologic symptoms. So that sort of brings me to the last part of the talk, and I wanted to, or I was maybe even a little bit instructed to, kind of hypothesize about some of the barriers and big questions left in DOHaD. And I really think that our understanding of these early life exposures and how they link to later life health outcomes, whilst we've made great strides, it's still pretty limited. And that's for a few reasons. The first is that DOHaD is complex. Our life is complex, and how we respond to our environments change over our life cycle. To date, most studies in our field have looked at single exposures, or even if they've looked at multiple exposures, they've looked at these in very narrow contexts or populations. And a lot of times, these have been looked at only through a biomedical lens. And so what this means is that we have no comprehensive list of exposures and their interactions and how these may predict risk or resilience trajectories. And also I think our kind of knowledge to action is limited simply because we're not doing a good job at translating or communicating what we're learning and knowing. So the dissemination of information in the DOHaD field to individuals, to stakeholders like policymakers has been limited. And this constrains the ability of these people to actually take what we've learned and act on it, to develop, apply context-sensitive and specific interventions. So what are some of the big questions that I think need to be answered or things that need to be considered? And this is not just from a research perspective, but maybe as we start to think about training the next generation of DOHaD scientists and clinicians. Well, we need to think about inter-individual and intergroup differences and how these really determine how an individual responds to an exposure. And fundamentally, we need to close health gaps. So that means that we need to do more work. In our field, a lot of us are working on women, but maybe we need to start doing more work on men. There's a health gap in DOHaD, and it actually has to do with men. But it also has to do with vulnerable populations, indigenous communities. So we really need to start broadening the approach of who we try to learn about, what's important to them to learn about, and how we work with all of these populations and groups and individuals to make sure that they are on the right start and the right path. We also need to recognize that an environment or an exposure that is suboptimal for one group or individual may be neutral or beneficial to another, all right? So it's, again, not just a blanket statement in terms of our conclusions that we can apply across populations and geography. We need to consider the contribution and weight of influence of different critical windows. Are certain periods of our life cycle or our development more important in shaping health trajectories than others? Well, surely this is going to be dependent upon the exposure. So the famine studies at least tell us that early exposure in the womb as a fetus seem to be more detrimental to later outcomes, but this may be different for different types of exposures. And we need to think about how the experience of a previous generation will then factor in shaping the health of a child in the next generation. And how do we consider our response as an individual and how that changes with age? So we might experience the same environment or adversity at one time in our life and respond one way, but respond completely differently later in our life cycle. And this is maybe, again, where epigenetics can play a role in helping us tease out these mechanisms. What about social determinants? Like I said, most of the studies have really looked at things through a biomedical lens. We know that social circumstances vary across the life cycle. They work with and they interact with biology to shape our phenotype, so we need to consider social factors. We also need to consider that social patterning for health and disease is different everywhere and for different people. So what might be an important social context and patterning in Canada or for a particular population here will be very different to somebody who lives in Africa, Australia, and even Northern Europe. And we also need to start getting a better handle on how much variation in our health outcomes do social determinants explain. And some people are certainly working on this, but we need to bring those people, I think, into the classic DOHaD realm because most of them don't realize they're working on DOHaD, and let's partner with them to have much more impactful and in-depth studies. And as I said earlier, I think we need to reshape our frame of mind. So what are the factors that actually provide resiliency? We need to stop thinking about pathology. We need to stop thinking about adversity. We need to shift our intentions to leverage what is good and sound and strong in an individual and group and allow people to flourish. So I think that's really important. It's really hard to do. It's not as sexy, but we really need to frame paths to resiliency. And then, even if we sort of think back on or currently on the evidence that we have, we need to look at it with a critical eye. How sound is the evidence that we currently have that links early life exposures to later life health outcomes? What is our knowledge base? We need to have a better idea of effect sizes and the variance explained. We need to do a much better job at harmonizing cohorts, and I know there are efforts even in Canada through the REACH consortium that are trying to do this. And this would also be related to harmonizing interventions. And again, we need to close those health gaps. We need to study diverse populations and vulnerable populations. I also think that animal models are super powerful to help us in understanding DOHaD, but we may need to rethink our animal models a little bit. They're very powerful in explaining etiology, and so we need animal studies to be harmonized more than they are, just like we're trying to do with human studies. We need to design our animal studies more like human cohort studies and RCTs. And we need to do a better job then at translating what we do in animals to the human situation. And finally, can we actually predict who is going to be on a path to risk or resiliency? And importantly, can we do this early enough to actually make a difference? Well, currently, there are no good tools that exist to do this. There are some tools, some risk prediction tools that are out there. A common one is the Adverse Childhood Experiences Survey, but a lot of these assume that pathways to risk are linear. They simply count risk factors. Many of these predictive models don't account for secondary challenges that an individual experiences. They don't account for interactions or weighting of different risk factors. So it's a very, very difficult thing to do, to put together a risk prediction tool or a resiliency prediction tool. But I think we have to direct some efforts to try to do that. And that's also because prediction can also help us explain things, help explain etiology, and it can also help us inform health policy. And sort of related then to kind of etiology and policy, do we have enough information, at least in some domains of what we're doing, to actually act, to intervene? Are we asking even the right questions? Again, this is where I think the community engagement is really important. What do the people that we're ultimately trying to help want and need? Are we asking the right questions that can inform policy? Do we even care about what policymakers want? Should we care about what they want? It's a really interesting question. I remember talking to Peter Gluckman a few years ago, and for all of the work that he's done in the policy field, he said something interesting, and he said that in the context of DOHaD, politicians don't really care about the insulin-resistant state or the BMI level of a child. They care if that child is emotionally and cognitively well. So again, are we asking the right questions that are going to get policymakers to act, to support DOHaD, to fund DOHaD, and the various things that we do? We need to make sure, if we're thinking about interventions, that we have really sound trial design. I'm not a trialist, but I'm trying to learn more about it. This costs a lot of money. It means I think we also need to move to having much more open data so that we can share and learn from what everybody's doing, and that will reduce the cost of burden in the type of work that we're doing. These trials, as well as just our other descriptive studies, need to be properly powered. We need to decide if we're looking at intention to treat versus a poor protocol analysis. And as I said, we need to consider what the participant or group wants. So if we're going to do an intervention, is this actually what the group that we're intervening on wants to do, or how they want it to happen? So, who is the intervention good for, and how do we shape it to make it good for them? And related to interventions, two very important things that I think are often missed. We need to assess safety in the work that we do. And safety is highly underrated and highly underreported, especially in DOHaD, where I think it's absolutely critical for legitimizing the work that we do, because we're working with highly vulnerable populations, pregnant women, young children, and babies. We also need to assess effectiveness and sustainability of interventions. So it's no good if something works in a very controlled trial state, but is this something that can be implemented and sustainable in that group or population or others when the researchers and clinicians leave? And what can help us determine effectiveness and sustainability might be the use of computers. So using simulations, using assisted or artificial intelligence and deep learning and machine learning. These can help us potentially predict the interventions that we're trying to design to see if they're actually the right ones we should spend all the time and money on implementing. And then I think thinking locally, I think as a community, not only here in Manitoba, but pan-Canadian, we need to ask ourselves, what are Canada's strengths in DOHaD? What do we want to be known for? And what's the issue, or maybe the top 2 or 3 issues that we should tackle? Is it obesity? Is it mental health outcomes? Is it improving the health and well-being of our Indigenous populations and our immigrant populations? These are serious questions that we need to have, because it will help to leverage and consolidate resources, I think, for greatest impact. So some of the emerging issues that I think are coming out in the field include, again, what's happening exactly in that periconception microenvironment. What are those conditions in normal pregnancy, but also in situations like assisted reproductive technologies? That's like the earliest type of programming, I think, and we still haven't figured that out. We're starting to learn a lot more about the microbiome. We've got experts in the audience here who do amazing work on it, but we still have so much unknown about the influence of the microbes on our health and development and adaptation to pregnancy. There are a number of amazing Canadian researchers who work on this gap in DOHaD: fathers, boys, men, and sperm. So I think this is an area in which not only needs a lot of attention, but we're really fortunate to have some of the best researchers in the world looking at programming from the male lineage. And again, transgenerational effects, super hard to study, take a lot of commitment, a lot of time, and those of you who do it, I think you're doing incredible work, and I really hope that more resources are devoted to looking at multiple generations. Interestingly, cancer is not something that we often talk about in DOHaD. We mostly think about non-communicable diseases, but this is a really emerging field. So if the young people in the audience want to carve out a niche, I think the programming of cancer is something that needs to be looked at and could have huge, huge impacts for the health of populations. And as I said, related to the example on flavor learning, we need to think more about the programming of our behaviors, not just what our genes do or our physiology in terms of measures of metabolites and inflammation. And then in Canada, as well as some places else around the world, we're now being faced with some very serious public health crises. And what do these public health crises mean for the programming of development and health in individuals? And specifically, I'm talking about substance use during pregnancy or even during the reproductive years, opioids and cannabis. All right, this is a field that we need to learn more about. And I think, as I said, one of the key kind of gaps that we're facing in really taking DOHaD knowledge to action is in our lack of translating and mobilizing what we know. I don't use that as a criticism of devotion, because I think you guys are doing some of the best KT and KMB work in this field, certainly in Canada. I'm really jealous. I wish there was an organization and group and capacity like this in Ontario, but not everybody is doing the job that you're doing. So we need to make sure that we are actually taking what we've learned and getting it to the right people. And this includes engaging with government at all levels, municipal right through to federal. And I've spent time on Parliament Hill, I know Megan has as well. Politicians like data, so we need to get good data and good numbers and communicate that in a very clear way to make them understand early life is a period in which we need to invest in, because it's going to give a really big return on investment for health, human capital, integration into society of Canadians. Irrespective of who we're talking to, we need to have consistent messaging. So I think that's something really important. Don't change the messaging based on the group. Have consistent messaging, and that will kind of like lay down almost that propaganda that we want to put out there about how important DOHaD is. And I think something that we neglect to do, because we often talk to ourselves, is going out and giving lectures. We really need to talk to medical students, nursing students, students in the education faculties, those who are in public policy and economics. Why? Well, in the case of these clinicians, these are the next generation of adult health professionals. We want them to know about DOHaD before they actually start getting into the clinic and treating these adults. We want them to know what's probably happened long before this. We want them to see children in their family practices and understand that they've got a really powerful position to influence health behaviors long term. Educators are really important, right? They're seeing the young minds, and particularly those in elementary and high school. This is a period when we can really shape health behaviors that can then get integrated into the home when the child goes back into the home. So these are important groups that we need to talk to. And obviously those who are in policy and economics, these are the people who will eventually be shaping our economic policy and our health policy and public health. And we want to convince them before they get into those jobs that DOHaD should be on their mind, and it's something that they should be targeting their efforts to in their field. So that's sort of the high-level summary, and I'll just take like 3 slides to tell you about my DOHaD journey. So I first started at the University of Guelph, where I did my Bachelor of Science honors in molecular biology and genetics and nutritional sciences. And there I actually kind of specialized my program into pregnancy and development. And that's where I got really big interest in what was happening in early life. And I was fortunate enough to then get into a PhD with John Chalice at the University of Toronto, and so he's arguably one of the founding fathers of DOHaD in Canada. He was the first institute director at the CIHR, Institute for Human Development, Child and Youth Health, former vice president of research at U of T, and former CEO of the Michael Smith Foundation for Health Research. And so at his core, he's a phenomenal DOHaD scientist, pregnancy and preterm birth expert, and I learned so much from him. And because of my interest in his, I was able to actually spend a lot of time during my PhD somewhere else, a tiny little place in the world that actually is one of the most powerful centers for DOHaD. And this is a tiny little place called New Zealand. It often doesn't show up on many maps. It kind of falls off the edge when someone takes a picture of the globe. But this is a great place. It's the home of world-class rugby, of kiwi and kiwi fruit. Please don't say that you're eating a kiwi when you're eating the fruit, because to New Zealanders that means you're eating the kiwi bird, and that's not a good thing. But I spent 6 or 7 times I went down to New Zealand to work at the Liggins Institute. And although I didn't work directly with Mont Liggins, he was long retired by the time I got there. I got to spend a lot of time speaking with him and working with some of the preeminent experts in DOHaD who live in this tiny little country. And that's me doing some of the fetal surgeries that I did as part of my, my doctorate there. So New Zealand's a great place. For those of you who like Lord of the Rings, there's Mount Doom in the top corner. So you can do major hikes on volcanoes, but you can also then go to exquisite beaches. And here's some other tramps, that's what we call them in New Zealand, hikes that we went on. I loved it so much I got recruited back there to work through the National Research Centre for Growth and Development and the Liggins Institute with Peter Gluckman, and also with 2 fantastic clinician scientists, neonatologists, Jane Harding and Frank Bloomfield, who also co-advised me during my doctoral time down there. And Peter, soon after my arrival, became the chief science advisor to the Prime Minister. So he had to take an arm's length approach to his work. And so I had great mentorship from Mark Vickers, who's really a giant in the DOHaD field with respect to animal models. And then Deb Sloboda came to New Zealand, and we did a lot of work together as well. And then at the end of my first fellowship there, I was recruited back to Toronto with Stephen Lai, who is one of the world's eminent scholars in preterm birth research, and he asked me to lead his group on DOHaD under his broader women's and infants research umbrella. And there it was great because I pretty much had a blank check, and I was able to really start to lay the foundation for the work that I wanted to do as an independent investigator, hoping I would get a faculty position. And I did, and that's in Ottawa, although I still have links with the Lunenfeld and Mount Sinai. I do a lot of my clinical research there, so we've got a big pregnancy cohort and obesity cohort, and I do a lot of work on the programming of neural tube defects there. So I go back and forth between Ottawa and Toronto, but you can go to my website to learn some more about our projects, but in general, very high level, I'm interested in this early life nutritional environment and maternal metabolic health and how that interacts with genes and microbes to really program development of the fetus and the placenta to set these health and growth trajectories that ultimately can have an impact in the short term, especially on neurocognitive function, but in the longer term, we're also interested in metabolic outcomes. We're also doing some work with HIV exposure in the womb. So again, I'm happy to talk offline with anybody about any of the projects we're working on. And I'll just leave it with a kind of final take-home message. I think we can all agree DOHaD is complex. DOHaD research certainly takes time, particularly if we're thinking about multi-generations. It requires interdisciplinary efforts, and it needs to do all these things to make sure that every individual has the healthiest start to life. And the burden of proof really is on us to do it well and do it right so that we convince people that studying and investing in the early life is important, even if we don't see what might manifest from that for 30, 40, or 50 years. So again, with this network and devotion, I really hope you're around for a long time. I'm jealous of the capacity that you have here. You're a real hub in Canada, and people know you internationally, so kudos to you. And I think because of the work that you're doing and what you've done, you know where we're going to get to. So thank you. And any questions I'll take. Yeah, so I think maybe that was captured in that post-birth time point that I listed, but absolutely. Again, this is the windows that kind of define who we are and what we're going to be, operate across this life cycle. And intervening, I think, first of all, whether it be giving glucocorticoids in threatened preterm labor or giving oxygen to a neonate who needs it, nobody's going to say, don't do that because that might program that baby 20 years down the line, right? The clinically indicated intervention at that time is important, but that's not to say we can't learn from what might happen to those individuals and then maybe adjust our care practice. maybe the sheer stress of the oxygen that we're giving is not the right one. We could still do something that improves or helps lung function or prevents preterm birth, but doesn't do it in a way that adversely programs to the same degree. So I absolutely think that that neonatal period is very, very important. Well, again, I think this kind of goes back to what I was saying at the beginning with definitions and what we want them to mean. That's up for us as a community to decide, and I would say taking a broader stance is a good thing, right? And you might choose to have your DOHaD period encompass X and Y, but someone else may choose to have it be Z plus plus, So it's variable, and it's something the community really needs to take a stand on. But particularly when we're thinking about interventions, then I think the narrower window of where that's effective is going to be in the earlier part of life. Yeah, it's difficult. I mean, I think you can take a page from the breastfeeding world and how they have to balance, promoting breastfeeding, but recognizing some women don't want to or can't, or just choose not to. I think it requires us to also have a really good understanding of what a healthy weight pre-pregnancy or weight gain is for one population versus another, because again, even dependent upon our genes and our background, we may be predisposed to have a certain type of body composition and weight or weight gain during pregnancy that may be perfectly fine for one group's pregnancy, but not so good for another. And I don't have an answer, but part of it is just having that sound evidence base across a variety of groups and individuals and populations, so that we can, yes, have consistent messaging, but also it needs to be context specific and sensitive. Yeah, again, people are rightly so learning to do studies not just on male rats and mice, but also females, and not just look at outcomes in boys, but also girls. So part of it, answering that, will be making sure we have sound evidence base now across the sexes and genders in a variety of different exposures and outcomes. Classic answer would be, well, hormones are different, and the way that a female fetus might develop in utero is different than a male fetus. And so that in and of itself, the sex effect is a mechanism that really needs to be teased out, but we need to look at it first, and everybody needs to be looking at it. And yes, it means our studies are going to be bigger and more expensive, but again, we need to close health gaps, and we can't just look at one group or another. So yes and yes. I think we've got some key windows that are obvious, not necessarily easy to work within. One of those is kind of children and adolescents, and just educating them on DOHaD. And there's some great work that comes out of Jackie Bay's group in New Zealand at the Liggins Institute, where they are educating high school students and younger on DOHaD and what that means. And even when I was there working with some of her initiatives, we would hear back from the students that when they learned about this and they took it home to their families started to make different choices at the grocery store and eat together as a family, and that can change behaviors. I think another key period, as I said a few times, is this postpartum period. So obviously, ideally, we want to predict who's on a certain trajectory and intervene very early. But if we can't, if something manifests, where is there a time that we still have a lot of flexibility to change the plasticity of an individual? And it may not be during pregnancy, right? It's hard to get interventions approved to be studied, let alone if we're talking about drugs. But there are other periods before pregnancy and certainly after birth that I think still have the ability to really, really shape the health and development behaviors of that individual. And certainly it will be dependent upon the type of exposure, but all of these exposures, nutrition, stress, those sorts of things, can be communicated in a way to young people, to children, to families, as sort of a holistic model of health and well-being. And I know that's making it very simple, because there are social and environmental inequities that make it such that you can't just say eat healthier food or go choose this versus that. But nevertheless, I think education at a really young age is one key window that isn't actually very costly, that we can use, that probably will have a really profound effect. And again, James Heckman's work has really shown that. And now they're second generation people that are followed up through some of his early intervention studies that really show the profound effects of investing in those early years. So how geography, for example, or life history may change someone's response to the exact same exposure? Is that what you're getting at? Oh, yes. Okay. Mm-hmm. Yeah, so pregnancy in and of itself is a stress test, so I think a woman is highly adaptable during that situation. And when we've got acute or chronic, really adverse exposures, it means that even in a challenge like pregnancy, there can be adverse outcomes. But when we're thinking about later on in life, I don't know if we have the same level of adaptability and plasticity. In fact, a lot of evidence in biology suggests that our plasticity goes down as we age. So our ability to respond to these stressors, these adverse exposures, the way they might affect inflammatory processes in our body, damaging our DNA, it's not going to be handled the same way in an older individual as in a younger individual or somebody who's also experiencing another challenge at the same time, like pregnancy. So yeah, there will be differences. I think a lot of that will come down to biology and cellular aging. It might come down to what our microbes are like at that stage, right? We're learning more how the microbiome, that population shifts in very old age from what it's like earlier in the life course. So it's complex, but I think a lot of us here are interested in trying to kind of prevent these exposures from happening, adverse exposures from happening, one, and two, if we can't, trying to mitigate their effects as early as possible. But I would be interested to see what people in gerontology think about DOHaD and how we can maybe learn from them, because to my knowledge, many of us haven't engaged with them before on this. No, sorry.
Welcome back, everyone. Hope you have enjoyed the lunch and had the chance to think and discuss a bit about DOHaD. The second half of the day today will feature two panels of invited guest speakers. We hope not only to highlight the DOHaD research currently being conducted in Manitoba, but also to help the audience envision the future of DOHaD and encourage capacity buildings in our province and beyond. DOHaD research spans a range of specialties, bringing together aspects of basic science, epidemiology, developmental biology, clinical research, population health, and offers opportunities for collaborations and innovative mixed method approaches to question. We have asked today's panel members to describe in 10 minutes how they interpret DOHaD, what lessons have they learned along the way, what they see as opportunities for future DOHaD work in Manitoba. We will open the floor for questions once the panel has conducted their presentations. Dr. Connell will introduce our panelists. Thank you, Kristin.
So I think most of these people don't require much of an introduction to all of you, but I'm really honored to kind of chair this panel and call up the first speaker, a phenom in DOHaD, early career researcher who is really making waves in the breastfeeding world, and also a good friend of mine, Megan Zad.
Thank you, Kristin. I don't think I've ever been called a phenom before. That was fun. All right. So this was our challenge to talk about these three questions. So I'll start with the first one of how I interpret DOHaD, which is maybe a bit different after the talk this morning, but I made the slides yesterday. So one thing, I think the classic DOHaD is about fetal development. So how is it that exposures in pregnancy can shape long-term health outcomes? But another that I've done some work on and I'm particularly interested in is about the role of the microbiome in developmental programming. So this idea that we're made up of not only human cells, but actually 10 times more microbial cells, and that these microbes have important roles in keeping us healthy, not just making us sick. And then I won't steal the other Megan's thunder, but thinking about beyond genetics and the expression of those genes. So I think really these kind of three images capture what I think about when I think about DOHaD. But also this, so the first 1,000 days, and for people who are maybe newer to DOHaD, that doesn't mean from the day you're born, but as we heard a lot about this morning, it's from the day you're conceived. So what's happening around that conception period, all throughout pregnancy, and then the early phase of life. So this actually counts out to about the second birthday. And what's particularly interesting to me about that period is that it's when breastfeeding happens, and that's what a lot of my research focuses on. So I think we also talked about this life course idea or life cycle idea and critical periods of development. So certainly that to me is also very important in DOHaD. So this kind of illustrates how we might have a particular fetus or embryo that starts out at high risk from the very beginning for some particular reason. Maybe it's a genetic trait or an exposure that mother experienced in the preconception period, and that really might put that fetus on a trajectory to non-communicable diseases like obesity or metabolic disorders. And conversely, you might have a very normal fetus who's on more of a healthy trajectory. But these are not set in stone. They can shift, and there are probably critical periods at which that can happen. I think one thing that's clear if you look at this figure is that the shifting, if it happens early, is relatively small. So maybe a small exposure or a small intervention could shift someone from high risk down to normal, or the other way around. But if you wait until later in life, it's not that you can't shift, but that shift all of a sudden becomes a lot bigger. So it can be more difficult to shift the curve later in life. And that's where I think the concept of critical periods comes in. So thinking about what might be modifiable, I think is really important to do. So as much as I love to understand genetics, we're born with our genomes and we can't much about that. But can we do something about how our genes are expressed? Can we maybe change our microbiomes? Can that start as early as during the preconception period? Or if we think right around birth, so I've done some work around method of birth, cesarean section, feeding in the first couple of days, so another critical period. And then thinking into that early postnatal period, so things like breastfeeding, but not just breastfeeding, so sleep during infancy, the mother's diet, the home environment, these are all possible things that could shift the curve. And then also thinking about later on, we were talking about that although it's scientifically and clinically interesting and important to understand the early life period, I think there's a sense of kind of doom when you say, well, that happens to a child and they're doomed for obesity. So understanding what can we do later on if we recognize that some harmful risks have occurred early on. So I do a lot of my own DOHaD work in the child study cohort, which many of you know, but is a pregnancy cohort here in Canada. Children, 3,500 of them recruited during pregnancy from 4 sites across Canada, taking a really broad view of development of health and disease, looking at many aspects with a big multidisciplinary team. And I won't say too much more about it, but maybe in the panel it'll come up. I think this is a really powerful model for looking at DOHaD. My own research, as I mentioned, focuses on breastfeeding, so looking for those epidemiologic links, but going beyond that then to understand, is there something in the milk, so are there biological factors we can identify, but thinking too about those social determinants or kind of outside factors that might be either contributing or confounding the relationships we see, I think is really important. A lot of DOHaD research is observational, so we have to keep those possibilities in mind. Thinking about mechanisms, so understanding not just that there is a link, but how does that link work, what's underlying it, and hopefully using that information to develop interventions. So that's my kind of broad overview of what I view DOHaD as in my own research. In terms of lessons learned, this was kind of fun to think about. So I think one is that DOHaD, at least from the human perspective, really requires longitudinal studies. So this is, of course, my favorite longitudinal study is the child study. This was last year at our 5-year birthday party for 1,000 children, so it was quite a day. But it takes a massive amount of commitment from these children and their families, and retention is so important when you're doing a longitudinal study, not only to keep your power up, so you need those big numbers to do research, but also for representativeness. So often the people you're going to lose first out of a longitudinal study are the people who are the sickest or have the kind of worst condition in early life, and those are the often most vulnerable. So keeping them in the study is so important. That's been a major lesson. And that retention really requires engagement on the part of the families. So we're maybe a bit behind the ball, but starting with some parent engagement committees in child, I think that's really important for a longitudinal study. And it really requires great staff. And I don't know if any of the child staff are here. They're probably seeing families, but they're amazing. So that's one of our Halloween parties. Some of the staff are in there amongst the families. And if you come to the lab, you'll see pictures all over the wall of these families. They really have a connection to the staff, and I think that's absolutely key to retention. So that's a big lesson in terms of human studies of DOHaD. To take a brief step outside of child study, I was able to go on sabbatical last year to visit another amazing cohort study in the UK. So this is the Ausback cohort, or they are known kind of locally and by the participants as children of the 90s. And they followed their subjects, their families. The kids are now, they're planning a 30-year follow-up visit. So it's amazing to go there and kind of learn from them how they've managed to keep going for this long. And it's not for you to read this chart, but like this was the first thing I saw when I got there, and I went, oh, because that's how many people like work full-time for this cohort. It's a big team. That's the org chart. It takes so many people, like they have a whole, data and patient engagement and the clinic, and like it's a massive operation. This is something I'm more used to seeing like in a company, but it's a huge operation. So that's a lesson, is that it takes a lot of people and a lot of money. I've definitely learned that. I don't know the solution to it yet, but keeping these studies going is expensive. I think other lessons, so when I think about challenges in DOHaD, one is this idea of triangulation. So making an observation in a cohort is one thing, but really understanding how that works and having people from different disciplines is really important. So in the child study, for example, we have environmental scientists, we have pediatricians, people from all these different disciplines are really important to have on board. Replication is important, so it's almost not enough to just see it in one cohort, so making friends with other cohorts is important. Consortia are really popular in DOHaD, and I think that's key. Getting to causation, so again, beyond the observational research. So this is where animal models, Kristin talked about earlier this morning, is really important. So having collaborations with people who do that kind of research can be very powerful. And even within the cohorts, there are advanced methods of analyzing cohort data with causal modeling and other things. So having statisticians as friends is also very helpful. And then the translation piece is key as well, of course. So having friends in the knowledge translation area, media, policymakers, all of these different stakeholders that we need for our research. And then this one. So I kind of drunk the Kool-Aid, I guess, on DOHaD and sort of thought everyone must believe this. And then sometimes I run into people who really don't. And I think it's important to talk to the skeptics sometimes. And the unicorn is there because someone recently said to me, this is like believing in unicorns. Like, you really think that something, happened when you were a fetus is affecting you 50 years later? And I was like, yes, there's a lot of evidence for that. But to them, it's just complete, they don't get it. So I think having that reality check for ourselves to really think about our questions, and is this what the evidence is showing, or is it our bias? And then thinking about how we convince those people. Messaging really matters. So I've had some personal experiences that we could talk about around the messaging of DOHaD research and results, and how it can either get misconstrued or can be really discouraging to especially new parents. And so I think we have to be sensitive to that. I think the field is really at a place, and this came through loud and clear to me at the last World DOHaD Congress. There have been a lot of cohorts, a lot of observations made, but the field really wants to move towards intervention. So what can we do with all these observations we've been making for decades? Can we please do something with it? So I think that's important, and HEALThy is this Canadian initiative. They're cohorts, but they're intervention cohorts, and I think that's where the field is really going. And then I think it's important to expand on prior research. So I'm guilty of sometimes seeing previous studies and thinking oh, cool, I could do that in the child study data, and just try and replicate it. And replication is important, but I think you have to also be thinking about new ideas. So what has nobody actually done before? And so when the child study started, nobody had been looking at the microbiome, but luckily someone was bright enough to say we should collect the diapers, and then it's been this huge field of microbiome research, but somebody had to be first and think about it. So that's a note for me to challenge myself to think about new things. And finally, opportunities in Manitoba. So this is totally stolen from Natalie, but I think devotion and CRIM are really like Disneyland for DOHaD scientists. So we have animal model researchers, we have cohorts, not just child, but the next gen cohort of gestational diabetes and others. So and we have links to policymakers in government. We have a Healthy Child Committee of Cabinet, which is, we're the only province that has that. So we have some really unique opportunities in Manitoba that maybe we take for granted sometimes and should not. So I think there are big opportunities. So I mentioned these cohorts, the administrative databases as well. So we have some really unique infrastructure on that front that can be used for DOHaD research, the Healthy Child Manitoba and the models. Those are the kind of the devotion pillars, really. But some other ones that I wanted to highlight is shameless, but I have this new Manitoba Interdisciplinary Lactation Center we're launching. So it's going to be a repository of human milk, lots of exciting possibilities, and data around infant feeding. The Center for Global Public Health. So maybe we don't have as strong as ties as we should, but there's a really great group of researchers there doing work in maternal child health in other settings around the world. I think we have a really awesome critical mass of new PIs right now. So several of them are in the room, but Megan Jones and Aisha, Salim and Karina Kowalik. There's just like a lot of new people here that bring a lot of energy, and I think that's something we should, if they're not already doing DOHaD, some of them are, but some are not. We just need to like bring them in and convert them because there's a lot we could do together. And we're recruiting right now 4 new Tier 2 CRCs, all doing different elements of big data. And we heard this morning multi-omics is really big in DOHaD, so we have to also convert them to be DOHaD researchers and bring them into our team. This is my last slide. We were asked about DOHaD opportunities in Manitoba, but I switched one word out and said for Manitoba because I think we can be players on the world stage. And so we have some links to, we heard about Heckman earlier today and his Nobel Prize and the importance of early life development. We have some links to his group. We have connections to the Life Course Epidemiology unit in Southampton. That's where David Barker was from. The LSPAC cohort. The other graphic there is an Indigenous cohort in the States. So we have these links. And this is from the last DOHaD Congress, some of the big priorities that came out, and I think we have the capacity to address them. So this idea of moving towards interventions, being multi-sectoral, having the translation into policy, and being part of collaborations and consortia. So some of those we're already doing, some I think we could do a little bit better. And we're hosting, in Canada anyway, the 2021 World Congress of DOHaD. So I think that's an opportunity for us to take a lead on this stage. So, thanks.
All of the speakers will convene at the end, and you can maybe jot down your questions now so you can ask them later on. But we'll move on to the next, and Megan 1 set it up really nicely for Megan 2, because I was going to say I think that there's an incredible critical mass of individuals here doing great DOHaD research and a lot of new PIs in the field, and I think this shows the investment and the longevity that Devotion and the University of Manitoba and CRIM are putting towards this area of expertise. And one of the newer recruits, but don't make that second guess her power as well. We've got Megan Jones, and some of you, because she's new, may not know her, so I'll give you a little bit more of an intro. She did her BS in biology at Mount Allison, then she went to UBC, where she did her PhD in medical genetics, and she is an expert in epigenetics, and she is an emerging player in, I think, what's going to be critically important to understanding DOHaD, how we can link the epigenetic changes and environments that work through epigenetics to health outcomes and trajectories. So, awesome. Thanks.
Thanks, everybody. I'm really excited to be here, and Megan, the sort of lead up to this, I think, is really perfect because we've already gotten such a fantastic overview of the important aspects of DOHaD from Kristin and Megan. I'm going to take us on a little bit of a different path, and I think that's one of the reasons that we wanted to do this today was to look at DOHaD from different points of view. So we've talked about population stage, we've talked about epidemiological data, but I'm a molecular biologist, a developmental biologist, so I'm going to talk about what happens inside your body and inside your cells and why I think that's really important to DOHaD research. So this is my sort of classic introduction slide about what I think DOHaD is. Apparently I lost my title. So we talked about this already, but an idea of, I asked the question about windows of opportunity. To me, one of the important aspects of DOHaD is this idea of a window of opportunity in early development where an environmental exposure is what we call biologically embedded. So this idea of biological embedding. And it links it to health outcomes later in life. So we have a number of, there's evidence from population studies from lots of different kinds of exposures leading to a number of different kinds of health outcomes. My research focuses more on prenatal exposure to inhaled toxins like cigarette smoke and air pollution, and their links to asthma and lung disease. But there's evidence from all kinds of exposures. And so the concept of biological embedding was originally, I believe, coined by the late Clyde Kurtzman at UBC in sort of the late 90s, early 2000s, and he was a sociological researcher who studied this idea of child, sort of adverse child environments and their links to long-term health. And his hypothesis, this is, remember, before we get into talking about the cells, his hypothesis was that something is happening in terms of the biology that specifically links these early childhood environments to later life health, but he didn't quite know what that was. So he termed it biological embedding. Now, as Kristin said, I'm an epigeneticist, so I'm going to try to convince you that the mechanism behind biological embedding is actually epigenetic. So here is my getting in the whole what is DOHaD. This is a way that I like to describe the concept of biological embedding and why it's so fundamental to DOHaD. I apologize to the Pink Floyd fans. I kind of feel this. Yeah.
I thought you were going to have a Pink Floyd.
So my concept of how we should be picturing these kinds of ideas to understand the mechanisms linking environments to health is if you picture your current state, your genome and epigenome primarily, but your developmental trajectories, all these things in common as a prism. And the environment that you're exposed to is a beam of light that hits that prism. So right now, this status, this particular exposure, combined with the current state of genome, epigenome, environment development, results in a specific possible trajectory of health outcomes. And this reinforces something that Kristin said earlier. This is not a deterministic process. Right? We're not saying this exposure plus this thing equals this spot. You are going or not going to develop a disease or not develop a disease. It's a trajectory of risk, but the idea is that you narrow those trajectories down a little bit, where your risk in those trajectories can be altered by your genome and epigenome. So the cool thing about the epigenome, and the reason that I think it's really fundamental to DOHaD, is because the epigenome is responsive to the environment. So the way we can add that to this is we imagine that at a certain period during development, in one of these windows of opportunity, your environment changes. So now this beam of light is hitting this prism at a different angle. So now we have a change in environment. What that can actually do is it can actually change your epigenome. Your genome itself doesn't change, like Megan said. Your genome is you're born with your genes and you're stuck with them, typically. But your epigenome can be changed, and it can be changed by the environment. So the idea here is that these alterations in the epigenome then can change your possible risks. So that's where we're getting at this idea of the epigenome being important to DOHaD. Because these are chemical tags, physical changes inside your cells that link an exposure to your health outcome. The other important aspect of this is that the epigenome is sensitive during certain periods, but at some point you can actually, once you get outside of those developmental windows of opportunity, even if your exposure stops or goes back to a different place, those epigenetic marks that are left behind at those specific developmental periods can stay. Not do stay, can stay. That's the important part here. Again, not deterministic. This is all about risk trajectories. So then if you have this example where your environment changes during a sensitive period, you get epigenetic marks changing, even after the exposure has gone, then this risk trajectory can be maintained for the rest of your life. And I use a specific example that I like. Remember, I'm a developmental biologist, so I talk about mice a lot. And this is one of my favorite examples of this. So this is the agouti viable yellow locust. Have you heard of this before? A couple people? So the agouti gene in mice is a coat color gene. It makes the mice brown. But there's a natural mutant that had a spectrum of coat colors. Remember, these are lab mice. They're genetically identical. But the mice were born with a spectrum of coat colors, from yellow at the top to more brown down the bottom. And what researchers discovered is that if you change the maternal diet during development, you change the epigenome at this gene in these offspring, and then you get different colors of animals. The specific nutrient they were working with here was folate, because one important epigenetic marker is DNA methylation, and it changed methylation at this particular gene. But this is a specific physical example of biological embedding. Maternal diet during pregnancy resulting in an outcome for offspring. Cool, nice visual, because brown, yellow, different coat colors, but like, who cares, right? Well, when these mice get older, the yellow ones get obese. The brown ones don't. They become insulin resistant, and they die earlier. Again, one specific macronutrient change during development in pregnancy is resulting in this lifelong change in genetically identical mice. There's no genetic differences between these two. Their genomes are different, but their genes are the same. So this is why I think that the epigenome is important, and that's why I do research into the epigenetics of DOHaD. So that's my sort of DOHaD thing. I decided to merge the lessons and opportunities together into one. So I picked sort of three main things that I think are important, and I want to talk about the lesson, and then I think the opportunity that we have here in Manitoba to overcome that challenge of that lesson. The first one is that human studies are messy. People don't do what you want to do. They're genetically, variable. They like to do things that, are outside of the realm of specific studies. So we have a couple of approaches to that. We have these beautiful cohorts like the child study, where we collect hundreds of thousands of people. The more people you get into a study, the more you're able to control for all these variability in studies. But the other thing, like both Kristin and Megan mentioned, was the idea of bringing in animal models. And I want to talk quickly about the enhanced study. So one of the two groups that came together to form the DOHaD is the biology breathing group, and one of the legacy projects for biology breathing is the enhanced study, where we are actually we're putting together an infrastructure and starting to generate animal models of prenatal exposures. We're starting with cigarette smoke. We're doing our first exposures about 3 weeks. But so this is something that we are doing in Manitoba to try to integrate the data that we're finding from human studies with controlled exposures in the lab. The second is that we still don't understand mechanisms. There's beautiful data linking environment and health, and environment at certain times and health. We don't understand necessarily why or how those things occur. That's where I think a lot of epigenetics comes in. But we also need to be doing more collaborative work to really nail down these mechanisms. And to me, if you picture that prism, there's sort of 2 areas we need to be looking at. We need to be looking at how the environment lays down the epigenetic mark. What's that connection? Then we also need to look at what's the epigenetic mark then do, and how does that self-fate? So there's these 2 really important pieces that we just don't really know enough about yet. But I think that there is a real critical mass in Manitoba that's going to be able to help us understand those cellular mechanisms a little bit better. And I think that the DOHaD group is going to really lead that charge. And then the final thing is the so what, and that gets at what we were talking about skeptics. Some people say, why do we care? You're born with your genes, you can't change your environment, so why does this matter? I mean, we're all scientists and researchers and knowledge users in this group, so we want to know because we want to know. But there are other reasons. So we need to start thinking long term about knowledge translation and mobilization opportunities, about bringing stakeholders in and helping them understand questions. I loved your idea earlier about getting into the classroom and teaching children about the importance of DOHaD and making sure that we start understanding from an early age that these decisions are important. And that the more we know about this, the better we'll be to make sure that every person gets the best start in life. I don't think I have a final slide because, this is, computer is about to die, just so you know. It's 1%. Yeah, well, yeah. So now we're done. We'll see y'all later. Okay, thank you. Yeah.
Okay, so as they're plugging in, I will introduce our last speaker from this panel before the whole panel comes up and is free to take your grilling. This is Alex there. I just met Alex, so hello Alex. I'll be interested to read your bio because I learned something about you. So Alex is an assistant professor here in the Department of Family Medicine here at the University of Manitoba, and it says you're an active clinician.
Like I saw patients this morning.
Okay,
As opposed to one that doesn't see patients.
Right. Well, no, I get it. I'm trying to make that joke. He teaches at the Family Medicine Center.
For the next month.
For the next month? Okay, just for the next month, so he can change his bio after that. And he's also interested in e-health, peer support, primary prevention, and research in policy matters, including electronic medical health records, data quality, and optimization of e-health. So I look forward to hearing this very different perspective on what DOHaD means to Alex.
I will. I hopefully will provide you a slightly different perspective, both as the only non-Megan on panel, and as a family physician, so I'm probably the only family doctor in the room, with the exception of one time one of our esteemed lecturers in the back once practiced general practice. So I'm an associate professor in the Department of Medicine, and I see patients, and I do some research, and I do some quality improvement work in addition to that. So to answer the question how I interpret DOHaD, I think I want to start by saying something that's maybe slightly provocative or maybe not. But I think, and maybe I should modify this line, but it's the clinical home. So the natural home of DOHaD to me is family medicine. At least the clinical home, because family physicians are the only specialists that actually look after patients from the point of time from which before they're pregnant, during pregnancy. We take care of babies and young children and adults and elderly people and people in palliative care situations. So this is the only specialty that actually covers both the developmental origins and the actual diseases. And I think that when you tried to explain, if Megan was to try to explain the sort of developmental, and either Megan, I meant if Megans would try to explain this to a family doctor, I think most of us would actually understand that this is absolutely true because we take care of patients at multiple life stages. So in some ways, I actually think that the natural home, despite the fact that my discipline doesn't have a tremendously strong research base the same way that, say, pediatrics does for various reasons, I think actually we're a natural home, and certainly when it comes to the knowledge translation aspects, we're really talking about primary care. If you're talking about the clinical realities of DOHaD, you're talking about primary care. Because once those kids hit 18, the pediatricians don't take care of them anymore. And when patients are pregnant, the pediatricians don't touch pregnant people. So I have a funny story. I was seeing the newborns. I see the newborns in the hospital once a week. And the mom had a health issue, she was bleeding, and I dealt with it, and then I went and told the nurse, and she was like, what do you mean you did that? The pediatricians never touched the mom. I was like, I used to deliver babies. Anyway, so the bench, as I see it in family medicine research, is actually the clinical data that our practices generate. We don't have an organ, we're not organ doctors, right? So our bench is the clinical data. It's our communities, it's our practices, and family medicine views itself as sort of a holistic, and we talk about the patient medical home and things like that. So the bench of family medicine is the clinic data, and so that's actually where much of my research lives, is in the use of clinical data, both for understanding patterns and observational type studies, but also now for some intervention studies. So we're doing some interventions, and we're doing them in a pragmatic way, so like what we call a pragmatic clinical trial, where we actually draw the data from the clinic. So we don't take people out of the clinic in a cohort or in a randomized control trial. We do the randomization within the clinic spaces, and we follow the data as it comes out pragmatically. So that's a real, I think, boon potentially to developmental origins of disease kind of research, because as soon as you take people out of the clinical setting, you introduce other variables that may not be true for the whole population, affect generalizability. And so the way I see DOHaD really is the same way that I see diseases in general, that they're part of a continuity from gestation to grave, right? When I see a patient with diabetes who's elderly, it's very obvious to me that diabetes is a spectrum of disease, right? I have diabetics who are on one drug and are going to basically live the rest of their life and not die from diabetes. And I have diabetics in my practice who have an amputated leg and are very troubled and have had diabetes for 20 or 30 years since they were 30. So that's a different disease, right? The 60-year-old healthy guy on metformin is a completely different person than the 50-year-old with an amputated leg who's had diabetes since they were 30 and is obese and has a whole other number of other things. So this is sort of natural to me as an exam physician. So I think that's how I see developmental origins, is that diseases are a spectrum. And I see all diseases on the spectrum, and we see patients who present early with disease when it's undifferentiated, and then later, after it's been differentiated and they're followed by various other specialists, they're still our patients in medicine. And so even that's the last line there, even the once formally diagnosed diseases, they're not even diagnosed yet for many of us. So we deal with behavioral counseling, where I tell people, you really shouldn't smoke. Well, the reason I'm telling them they shouldn't smoke is because I'm trying to prevent them from getting some disease down the road. And I think it's also natural when you think about, I really like that spectrum that Megan showed before, because sometimes the residents have a very hard time understanding how we can have a 9-year-old in our practice who smokes. Like sometimes someone with genetic dice worked out really well for that person, and they smoked their whole life, and they didn't get lung cancer when they lived into their 90s. And that's normal to me. I have a few, I don't have any 9-year-olds, but a few 80-year-olds who smoke. So you're not doomed to get lung cancer because you smoke. You're just more likely to. And so the more I think about the research that DOHaD and the sort of clinical application, I go back to the sort of statistical probabilities. It's a range of diseases and a range of likelihoods. So I think clinically that's for me how I see it, and as a family doctor that's kind of natural. So this is the devotion slide. My research really lives in that sort of administrative clinical data. I'm not a bench scientist, even though once in a past life, many years ago, I was the person who got to kill mice and harvest the spleens, but that was in my undergrad. I don't do any more technology, so I don't. If you ever need someone to harvest spleens, though, just for fun, it'd be, given the current politics, it's actually really fun. I'd love to have an interest in killing some mice. Anyways, so what lessons have I learned along the way? I think the most important lesson for me, as someone who's a clinician, who started as a clinician first, but became a researcher or a clinical researcher after, was to let the data, use the data to tell a story. And that's, I got into research trying to tell people, trying to tell doctors to care about their data quality. That was sort of the first research project they did. And I found out very quickly that when you show a data set to a bunch of doctors, the first thing they tell you is, oh, the data's wrong. The data's not right. Whenever you show doctors their data, that's inevitably what they tell them. And so I sort of evolved my thinking around this, and I said, look, the data is not perfect, and no data is perfect. Whether it's from a randomized controlled trial or a really well-done cohort or whatever, it's not perfect, but it tells you a story. And so what I've tried to do in some of the work I've done recently is to let the data tell us a story, and I think that's an important lesson for DOHaD researchers as well, is like, what is the story you're trying to tell people? What's the, that's this idea of knowledge translation really boils down to letting the data tell a story and using the data to provide a message, whether that's we should clean up the data, or that there's some really interesting findings that lead us to other findings, or new research or interventions, and I think that's important. The other piece, this is maybe a counterbalance to Megan's saying, clinical human trials are really hard because humans, don't listen, and we can't just sacrifice them so that we can harvest their organs like we can with mice. But clinical data is incredibly valuable and requires, as Megan number 1 said, a tremendous amount of effort being time and money to acquire and curate particularly data for secondary use. So I use clinical data for secondary purposes. It was collected for the purpose of providing care, and then my research with my shoestring team tries to curate that data and clean it and code it so that we can then use it to link with other data sources or to describe patterns of disease and things like that. But that takes a lot of effort. It's not as easy. People imagine like you just, the doctor puts it in the computer and that must mean it's all useful. It's not. It actually takes a lot of effort to curate it and to make it well, and that's probably why we're hiring 4 big data scientists to try to help us with that. And part of that cleanup and curation is really understanding the inherent limitations. If you understand the inherent limitations of the data that you're using, and you understand what it does tell you and what it can't tell you, then you have a better, to me, that's the lesson that I've learned in terms of we can then actually achieve the goals that we want. So, a few years ago when Megan came and said, there must be breastfeeding data in primary EMRs, I said, absolutely there is. But it's not all there. And we don't have any clinic that can run. And there's a few little limitations that are going to make it challenging for us to acquire all that breastfeeding data. So maybe if we get a third of the province's breastfeeding data, is that okay? That's an inherent limitation. We don't have the whole province. It's not, but it is a representative sample. A third of the province, or even a quarter, is probably okay. And so maybe we should just focus on that and understand that even within the data that we collect, because it's collected for clinical use, there's going to be some messiness. It's not going to be perfect. But if we know what those limitations are and we can describe it, then our story will be accurate and we can be happy with it. So that's one of the lessons I've learned. This is just an example of sort of messy data that we turned into an interesting study. I won't belabor the point too much because probably Liz will get up and yell at me. But this was a study that we tried to develop a case definition for diabetes using primary care data in children. We actually set out to distinguish between type 1 and type 2, and we weren't successful at that, but we were able to describe a population. And then the story we've been told with the data, this idea of using the clinical data to tell you a story, was we found within these data set, within this single data set, we actually had ranges of definitions. So we had this really tight case definition at the bottom, but then depending on how you use the clinical data, depending on which variables you included, all of a sudden the number of patients who potentially had diabetes changed, and it was changed fairly dramatically. And then the other thing we could show was that this range of diagnoses, so within the lab data, within the A1Cs, many of the kids that we found, almost 60%, had a pretty low HbA1c. They were not terrible diabetics. They were actually diabetics because this was primary care data. These were diabetics that were kind of living in the community, doing okay. The diabetics that the pediatric endocrinologists see, they're these guys. They're the ones that have bad A1Cs, or they're the ones that they shouldn't be seeing at least, right? Maybe they can get them into control eventually, but and then there's this kind of middle range that's a bit out of range. So it tells you something about who is this population. Actually, most of the pediatric diabetics had pretty decent control, but a pretty important sliver had terrible control, and those are the kids that, in that narrowing epigenetic spectrum are probably much more likely to get disease. So if you're going to focus an intervention, who are you going to focus your intervention on? Are you going to take all pediatric diabetics? Are you going to take, this slice or that slice? So trying to figure out, letting the data tell the story and figuring out what you want to do with it is, I think, a really important piece. So what opportunities are there for DOHaD research in Manitoba? This is my clinical world. I do data stuff, and I'm a data scientist, and I like to use technology. And then I go to my office, and I feel a little bit like, Mr. Scott, who's like talking to the mouse, because we are in clinical practice, like 20 or so years behind pretty much every other industry in terms of how we use the data that we generate clinically, more or less. I mean, there's spheres of places that do things really well, but I mean, the fact that we still use fax machines and pagers is evidence that we don't actually use data like other industries. Like, if you go to your bank, and just imagine for a second, right, that you went to your bank and handed them a piece of paper and said, please give me some money. They wouldn't give you the money. They would say, give me your card, give me your PIN. You could actually walk across the street to any bank, and then they might give you money, or unless you had account. But, and you can go across the street to another bank with that same card from a different bank company, and somehow they can talk to each other. We actually can't really do that at large part of medicine, right? We hand people pieces of paper and say, go off and get something done, get this lab done, or whatever. So the clinical data that we generate is still very antiquated. So there is a tremendous opportunity though for data linkage in Manitoba. So we need to acquire more data, I think, and we need to acquire it in a modern context, and we need to link it better and more so that then you can start to triangulate. Because each individual data set has its own inherent limitations, the more data sets you can layer on top of each other, the more actually you can describe things that are meaningful. And so we need to sort of advance this science. I think it's really urgent and important, and bring it up to the sort of standards of what modern technology companies and banks and grocery stores and airlines, how they handle data. And if we started handling data that way in medicine, we might get a lot closer to actually really understanding some of these developmental origins. Because if you walked down Portage Avenue into your Canada building and asked them, how many people were on a plane at 1 o'clock today in Canada, they could give you that answer like within minutes, right? Someone could go in the computer. And if you said to anybody in Manitoba today, how many people saw the doctor today, that is not an answerable question. That question is not answerable today. In like 6 months from now, or a year from now, Manitoba MCHP could tell you the answer to that question, but not today, right now. Okay? So this is again, this data looking retrospectively from a study that we did that looked at weight and growth and height measurements. So this is clinical data that shows, we have pretty good capture of any measurement. Getting all three, height, weight, and head circumference, not as great in 1 month, and then not surprisingly, in the sort of 1 to 3 month period, we're pretty good, and then it slowly drops off after that. But so if you're talking any measurement, we've got lots of data. There's more data around growth, heights and weights in the database that I have in Manitoba. Well, at least the Canadian database, I'm not sure about Manitoba, that has been collected in terms of heights and weights by StatsCan in its entire existence. So there was a study that the group that I work with published in 2015, and we looked at obesity trends, and the obesity trends that we described were based on height and weight data that was greater than all of StatsCan's height and weight data. So this is just pediatric data, this is just Manitoba, but it shows you what our capture looks like. Lots of kids are getting weighed, less of them are getting the heights and weights and head circumferences at every visit. And we have this data. It's there. It would be great, I think, a great opportunity to link it with other data. This growth data is super important, right? Because growth tells us lots about other things. So it's just an idea. And then I go back to this sort of what would Google do? How would Google being the best manager of data, at least the most successful thus far? You can maybe argue Apple, but somewhere between Google and Apple, probably the best managers, the most successful managers of data out there. And this is how Google handles its map, right? You think of the way Google Maps works, right? You go and it's layers, right? You can add the satellite view, or you can take the satellite view off. You can do a little world thing and switch around. So think if we did this, what if we did this, or if Google was doing this, if we had that level of data science, we could do this for the sort of epigenetics map, or the genome to social determinants map. And you can actually look across the spectrum. So you can pick something here, but then go up or back or down, satellite view, layer some other things on, show me the topography, show me the street level. That's the kind of thing that if we could do that with medical data, the same way Google does that with your personal data, to just so that they can market you the exact right product, right? Because it's astonishing how good they are, right? Even though people we're really smart people in the room, I'm sure we are. We don't fall for marketing. But you fall for marketing stuff every single day because you buy stuff. And so whether you know it or not, you're being marketed, you're being tracked, you're being followed, and Google trades on your information. And they use this sort of very discrete descriptive at aggregate levels to market you appropriately. And that's why when I eventually am lucky enough to buy Tesla, I will have been successfully marketed because every day on my Google phone I get articles telling me how awesome electric cars are, why I should buy one. And I'm like, geez, Google, you know me so well. Anyways, I'll stop there and let us join the panel and take this thing off. Thank you. Here's the mic.
Okay, I'll put the mic up at the front in 1 second, but does anybody have any burning questions as the panel settles in? I'm going to call on people even if they don't put their hand up. Okay, you can start.
So one thing we've heard today, it's a good thing, is that DOHaD is not disease specific, it's not specialty specific. And that allows for collaboration. On the flip side, I think it often means that we're doing this research off the side of our desk. It doesn't really, in this institute even, we have Devotion, but it doesn't have a platform, it doesn't have a recognized academic program. And I'm wondering how we go from this broad approach to giving this the legitimacy that it deserves. So I'd like to hear your perspective on that.
So I'll just repeat the question. If I paraphrase incorrectly, let me know because this is being recorded. So the question has to do with how do we go from doing the research that we're doing, sometimes in disparate ways and contexts, to actually having a platform with which we can really mobilize training and actually put things into action.
Well, I think today is a good start, so having some education and training specific to DOHaD, and I think we could brainstorm about what the best way to do that is. So whether it's through symposia like this, where people kind of have to sign up and come, we have to market them and get them out here, or whether we want to embed it in existing training programs, so the medical school, the nursing school, the new pediatrics grad program that may be starting, going to undergrads. But I think we as Devotion don't have unlimited resources, but we could be a champion for that. So go out and connect with the existing training programs to make it a more official part of what's going on. So that's one idea.
I'm going to get this program name wrong, but the Bachelor's of Health Sciences, I think, program is being revamped right now at the University of Manitoba, and the discussions that I've been part of are, there's already pieces of DOHaD in that degree. They're going to do a little bit of a focus on social determinants of health, like that. And so there is a discussion of already potentially building some of that into a couple of different programs. I also think that days of today are great, and I also have questions earlier about going out into the community and you know, speaking the truth of DOHaD to the world.
The unwashed
masses.
Teach the unwashed masses about DOHaD. No, I actually, or unicorns. So I actually think part of this is figuring out what's the right infrastructure. So, you know, I mean, if I was king for a day, we'd hire a whole bunch of really competent people, house you all in the Department of Medicine, and first we need competent people, which we don't have, but, and make this sort of a program of research within sort of an institutional framework, the same way there's a CRIM, there would be some kind of, you know, functional framework. And, you know, truth be told, more people give money to sick kids than do their family doctors, so it probably will live inside pediatrics, and that actually makes a lot of sense because of the momentum and the sort of the momentum that this institution has around pediatrics. But to me, it requires some form of infrastructure so that, you know, like the Cardiology Institute at the place where I work, which is sacrosanct because, you know, lots of people have heart attacks and give money to the Cardiology Institute, there needs to be sort of a similar kind of infrastructure around, you know, those labs should be co-located and those people should work together and have symposia like this, and there needs to be some kind of infrastructure that ties that together, regardless of the umbrella. Like, I'm being facetious by saying it should be family medicine. It shouldn't, but it probably needs some formal infrastructure to really succeed in the same way that, you know, cardiology research or nephrology research has done well in this province.
Other questions?
Great.
Yeah, I saw Hippocrates doing on the screen earlier, and I think one of his famous saying was that let food be your medicine. So if we are going to look at the developmental origins of disease and food is not prominently featured as well, even just capturing data on what are they eating, apart from just milk, combining all that, that would be a very good data set for us if you already have such a big number of kids who are already participating in the program.
So you're 100% correct. And whether you know it or not, the data on what you eat for dinner exists. Assuming you use a credit card to buy your food or have a Superstore points card or an Air Miles card, basically, you know, people get worried about Big Brother, they're like, I don't even know what you're having for dinner. They already know. Like, your grocery records are out there. They're just not linkable. So, you know, between Air Miles and the points, the Superstore one, that's probably 80-90% of the grocery store purchases. So, okay, we'll list saying Neons or whatnot. But at the end of the day, that data is there. It's just a matter of how do we acquire it.
I think that's interesting to hear you, the data guy, say this to the nutrition department, because I know we have food frequency questionnaires in child study, and I already get told, well, food frequency questionnaires, that's not a dietary recall, so it's not as good, and so jumping this gun. But I think, like, it's happening at Outback when I was visiting, like, that was the next data set they were going to link in was loyalty cards from grocery stores to get that information and see what people are buying. So I think those are two separate issues, but I agree, nutrition is a huge part of DOHaD. It's kind of where DOHaD started, right?
And wouldn't that be a really interesting intervention? You know, when I lived in New Zealand, my
Food Town loyalty card would send me these emails because it knew that I purchased a lot of apples and vegetables and Brussels sprouts, and then next week, all of a sudden these things were on sale. What about having an intervention where those emails are then shifted towards more sale on healthy foods?
You've been ordering a lot of chips.
Yeah.
Maybe you'd like to consider potatoes.
There you go. I saw some other hands up.
It's interesting that they're doing this in the UK, which just further demonstrates my point that they're 20 years ahead of us.
Yes.
A question in terms of the perspective of DOHaD when it comes to going to antecedent generations. The meaty data and the F1 consistency of epigenetic change is true to, at least the female to the daughter. Why aren't we looking at least grandmothers of the kids? And should we be undertaking to encompass that component in DOHaD?
So just to repeat and slightly paraphrase, are you specifically asking in child, for example, going back to grandparents from that cohort, or just in general, looking transgender?
I know why we're not doing it in child. Way too expensive.
Yes.
But in general, the concept.
Well, I think we would all agree, and I know, Alan, you weren't here this morning for Kristin's talk, but that was something that she pointed out as well, which I think is really important. It doesn't start just from when you're conceived, it starts way before that. So the multi-generation cohorts are really important. That's another thing they're doing in ALSPAC, so they're going and getting all the grandparents enrolled and asking them questions, so I think it's a really important perspective.
Yeah, and in terms of, for the epigenetics in particular, intergenerational transmission of epigenetic marks is fraught and highly controversial. But you don't need to inherit specific marks in order for this to be epigenetic mediated, right? If you get prenatal programming that results in epigenetic marks in grandmother, that results in a specific exposure in utero, stress hormones is the classic example. So intergenerational transmission can happen without actually inheriting epigenetic marks, and it doesn't mean that epigenetics is involved. I agree, we definitely need more cohorts that are doing intergenerational work. It's one of those opportunities where animal models are going to be beneficial, but while human studies, I did say human studies are messy, I mean, animal studies are very limited. So we need to be able to bring those two things together.
And I wonder if even flipping it from the other perspective, and cohorts that exist in like aging populations, can we go to them and then ask, can we recruit your children and your grandchildren and learn some things about them?
So part of the answer to your question, you answered as you were asking it, because you have to have written, I think, a grant for the child study, which is, it's really expensive. So going to the grandparents becomes really expensive. So then the question to me is, how can we acquire some of this in less expensive ways? Is that using clinical data in other ways, or other types of linkages? Because unlike mice, humans live a really long time, and so to follow grandparents to their kids, that study will wrap up in 80 years, and then we won't be here to actually publish it. So, you can get 4 generations of mice in a few months, but you can't do that with humans. So we have to figure out another way that we can do cost effectively.
And I like what you were saying earlier about harmonization, cohort harmonization. I know that's something that came up at some point in the child study, but what we really might not know is the CLSA, so the Canadian Longitudinal Study on Aging, is recruiting an older population. Chances are good that some of those people are related to the children in the child study. Wouldn't it be great if we could link those things together?
Yes?
I have a question for the first. So you were talking about the Manitoba International Lactation Center. So what exactly is that like section of breast milk?
So the question is about the milk.
So just one answer to make, I'll give you the real answer. It's the best acronym that I've heard in probably years. Since child.
So is Lauren Hillier? She's not, because she even thought of the acronym. Manitoba Interdisciplinary Lactation Center. It's new funding we have to set up. There's 2 halves to it. So one is a database to collect infant feeding data. So my colleague Nathan Nickel from the Center for Health Policy had started a feeding database because Like Alex said, I went and said, surely we must have collected this data. Turns out, kind of not really. There's data from the hospital that are pretty good if babies are breastfed in those first couple days, but after that, not well captured. So Nathan has set up a system where when moms are bringing their babies for regular visits, they fill out a little form about how they're feeding their babies. That was a grant that was ending, so this new center will continue that and expand it to more locations in the province. So we're going to have province-wide data on how babies are fed, which is awesome. It will let us do things like link that data with growth data, see how it's affecting growth, or allergy data, or test the impact of intervention. So some of you may know in the last couple of years, lactation consultants were cut from the hospital here, so we can see, did that have an impact on breastfeeding rates? And then the other half is a biorepository, so when those moms are filling out their questionnaire, we'll ask them, and if you're breastfeeding, would you like to give us a little sample of milk? We'll bank that in a biorepository. It can then get analyzed down the road for different components, and then we can link that with maternal data, so if moms have different health conditions, is their milk different? And we can also link it with the infant data, so our different components in milk influencing babies' growth, and reviewing it as a platform so that other researchers who are interested can come and access either the data or the milk or both. Some of the first projects we're going to do are going to look at medications that are passed into breast milk, because it's one of the reasons that moms worry about stopping breastfeeding early, so that's one example.
I just have a question about milk. So, say if like one mother is healthier and like had less negative epigenetic effects, like would there be benefit to then giving her milk to a different child and to that child?
That's a fascinating question. We actually have a student this summer together, so we're working on this. We're like, epigenetics, breast milk, we should do something together. So we're going to look in the child study if breastfeeding influences epigenetics in babies. You're getting like a few steps ahead, but yeah, this idea of can we optimize breast milk composition somehow? Can we maybe give a certain supplement or dietary recommendation to moms for them to change their milk composition so that it's better? We also are planning to look at things like donor milk, so for babies, especially preterm babies whose mothers can't make milk, they're getting donor milk, and how can we make that the best way possible? But I feel like we could talk later a lot about breast milk.
There's a fascinating evolutionary biology link to this. If you think about it, which is we in current modern society think that someone else breastfeeding the baby, which is just like abhorrent, right? But historically, from an evolutionary point of view, that was totally normal. To have the wet nurse of the community who breastfed all babies, and and moms went back and worked in the farm or whatever, or the fields. So if you think about this from evolutionary biology, sharing milk is actually totally normal. We've decided it's not something that's normal in modern society. So it might actually be beneficial in some way, or not. Figure that out.
Okay, we now need to move on to the next panel. That went really, really fast. Thank you so much. There may be time at the end for further questions from everyone. Okay, so the next panel is going to be kicked off from the neonatologist perspective on what DOHaD means and lessons learned. So welcome.
Thank you.
We've got Dr. Dakshinamurthy, and she is in the Department of Pediatrics, does some amazing clinical and translational research. I first met you at a CNPRM meeting a couple of years ago, so I was really excited to see your name on the program, and I'm interested to hear what you think about DOHaD from your clinical perspective, but also how that can relate to some of the research that we might be able to do in learning about mechanisms, interventions, and giving babies the best start to life.
In a way, I'm a bit of a DOHaD interloper because I actually don't do clinical research in the newborn. I do research in animal models of catastrophic disease in the newborn. And so in a way that what I'm going to talk about kind of in a slightly punchy post-call kind of way is going to get at that. And it's really the difficulty of finding a way to place catastrophic neonatal illness into the context of DOHaD, which we all know if you have a bad start, it flashes forward for the remainder of your natural existence. But how do you capture that in any way that we can examine as a component of DOHaD? And just to illustrate that, I'm going to talk a little bit about my research only because I want to give it a bit of context why I'm never quite sure whether this is DOHaD or not. So I'm working on persistent pulmonary hypertension of the newborn. This is the alphabetology of neonatology. Everything has an acronym. Pulmonary hypertension of the newborn is relatively common. It complicates about 10% of NICU admissions. And if you think about, there are 55 NICU beds in the hospital here, counting all levels. That's a number of patients at any given time. And the causes are multifactorial. Some of them occur before birth, some of them occur during birth, and some of them can occur right after birth. But generally it occurs within the first few days of life. And the big thing that has to happen in all newborns as a part of their developmental necessity is this thing called circulatory transition, where you go from being on full life support courtesy of mom to you're on your own, do it yourself. And you're on your own, do it yourself involves a huge transition of the direction of blood flow from blood going to the placenta to blood going to the lungs, and up till birth the lungs don't require blood, don't get blood, and then suddenly they're getting their entire cardiac output. So this is a plumbing problem which becomes a physiological problem. It also complicates a number of other diseases which we do recognize have DOHaD implications, because we see some of these problems showing up in traditional DOHaD fora. These are arguably the sickest babies in the NICU, and honestly, this is why I was interested in this disease. I'm interested in catastrophic illness, because if you look at stakeholder groups and have a conversation about what diseases are important to them, there's one population that's missing from those groups and we're a little bit blinded to. And those are the populations of non-survivors. If babies don't survive the NICU, they are not, their parents are not in stakeholder groups later. And so if you're looking for what makes the greatest impact on developmental origins of disease, I think we do have to open our eyes to the vanishing population that does not have a voice there. So catastrophic illness, high mortality, very fastly progressing, probably the fastest progressing of all the vasculopathies. You really only have a week or two. And if you manage to take advantage of that limited time window and the patient recovers, there are some lifelong clinical implications with respect to cardiac pump function, with respect to response to hypoxia later. I don't recommend any of these kids go on hypercritical altitude. They certainly wind up being a huge population of the pediatric ICU. With every intercurrent illness, they get catastrophically ill. So there is a survivor component to this. But this is a question that I get a lot. If I have a conversation about this in the DOHaD forum, because really this is not DOHaD. This is not traditionally considered DOHaD. And so in being asked to present on this panel, this was my dilemma in saying yes. And this was also my dilemma I want to talk about with you guys, because you get a really nasty disease, you either die or you get better, and maybe you have complications later, but we really don't know how to capture that. And there are very important reasons why we don't know how to capture that. Well, I can actually tell you this about any value whatsoever. This is part of the problem. You can't read it on the bottom. It says, late at night without permission, Ruben would often enter the nursery and conduct experiments in psychoelectricity. There's a limit to what you can actually do with the NICU in studying pulmonary hypertension, and in fact in studying any catastrophic illness in the ICU setting. So he says it's a mammoth. We have to start somewhere, so therefore animal models are the way almost all of this research is conducted, including my own. What I've learned, which is the question I was asked to speak to, well, the choice of animal model that we use really determines how we view the disease that we are studying. And I would put it to you that, yes, accuracy of disease modeling is entirely dependent on your choice of animal model. and how well you can generalize from that animal to the human. I was in a pharmaceutical drug development meeting, and what I was told, the tagline was that mice lie, rats cheat. And this is the point, because when you're generalizing data from rodent models, it's always known that there are such huge physiological differences that you may fall into a chasm in between this very relevant research and the human model to which you're attempting to apply it. But also, when discussing DOHaD, by definition we're talking about longitudinal work. And longitudinal work requires survival studies. And survival studies require that you can A, afford to, B, manage to, because you have to feed that animal, and in neonate, that might actually be quite a bit of nursing care. And then survive that animal. And then get it to a later point in its lifespan. And the larger the animal, the less likely it is that its lifespan is a manageable length. But we're not now talking about 3 generations of rats in a few months. We're really talking about quite a long period. And then the very practical details of how big are they, where are you going to house them, what do they need to eat, and can you actually consider keeping them alive for long enough to make any kind of inference about the longitudinal developmental effects of whatever catastrophic event that you had in the neonatal period. So I'm using a postnatal hypoxia model, and happens to be piglets. They're actually the same size, really, as a human baby. The development of the lung is at the same stage as the human newborn, which is absolutely necessary for any studies related to the lung, except for the fact that by convenience, about 50% of the studies on PPHN are actually conducted in rodents, which are born in a very completely different developmental stage, have preterm lungs, and really don't make good disease models of neonatal disease in this case. If you want to look, however, at survival studies, rodents are really good for that. If you want to look at antenatal effects, I'm not messing with a 1,000-pound sow. I'm not going anywhere near her. I will wait for the farmer to hand me the piglet at birth, after 8 hours of colostrum. But that doesn't tell me anything about antenatal effects on that animal, nor can I model them in any useful way. Lab studies are ideal for that, because you actually can house the youth. They will have a categorical number of 2 labs in there. You've got your control and your test. All the studies can be done on one, with the other one being the control animal. You can do a lot of antenatal work, including closure of the ductus arteriosus antenatal. What we understand about the antenatal effects of maternal stress on neonatal weathering transition has been done in lambs. Incidentally, lambs only have, lambs are born in the spring and the fall. So you're obliged to do all your experiments at those particular instances, and then you have a fallow period where you get to write up. That's not very practical. And so I would say in PPHN research, we're kind of divided among the piggers, the lambers, and the ratters. And the ratters are the only ones that are considered DOHaD. But I will point out to you that there are a whole lot of reasons why that is an animal choice of convenience that allows you to enter the survival study forum and thereby talk about long-term effects of what happened. So I'm going to conclude by saying, since I think you should be a little bit more explicit here in step 2, is that a miracle happens. Almost everything we do, we look at one time point, and then we're trying to extrapolate to time points way down the pipe. And neonatal stress and anything that causes catastrophic neonatal stress is a one-time point blitz, and then a whole lot of life happens. And then we're looking at effects in juveniles and in adults. And our ability to relate these things is limited, very limited in non-survival animal models. Even in survival animal models is very limited. And our ability in humans to infer causality is fraught, I would say, and much more fraught than a rodent living twice. So when the last question asked me to reflect on what can we do to enhance DOHaD research along these lines, I would say, if you want to look at the effects of neonatal stressors on lifelong consequences, it has to be a combination of appropriate experimental work in an animal model you didn't pick simply because it was cheap enough to survive, with population-based studies that would support that and specifically support some of the linkages and causality that you can really learn in animal models.
I love sheep studies, but I'm biased. So our next panel speaker is Dr. Robert Schwab, and I'm really happy to see somebody who's doing dental sciences here. I think that this is definitely an under-researched area in the context of DOHaD, so I'm excited to hear what you have to say about what DOHaD means to you and what we can do as a field to improve oral health. And so you co-lead the Early Child and Health Promotion and ECC Prevention Project in Manitoba. I'm not sure if you're going to talk about that at some point, but that would be really interesting to hear about as well. So everybody, please give a warm welcome to Robert.
Thank you. Thanks so much. And so I think today I'd like to share with you a little bit sort of answering the three main questions and then sort of giving reference to some of the studies that we've been doing to sort of back up sort of my comments about sort of whether or not oral health and my early childhood oral health research program fits into the DOHaD theme, which I think it does. Although recently someone had asked me, does your research fit into DOHaD? And I tried to explain yes, and they sort of said, no, we're not worried about sort of oral health of young kids, but what about later in life? So I think this is one of the issues that sometimes people think with DOHaD is the outcomes for health and disease don't necessarily have to be 30 or 40 years away, but they could actually be within the toddler years or the preschool years as well. So how do I interpret DOHaD? So when I think of DOHaD and oral health, I particularly focus on dental caries. So that's sort of my little niche area that I've layered in on over the years, and particularly decay during the preschool years and infancy, which we know is quite problematic in Manitoba. And I sort of give credit to my past mentor, who's a pediatrician, Dr. Moffat, who really got me started in this area of looking earlier on in kids' lives to look for those early origins of risk. I think as well the concept sort of that came clear over the course of my graduate work is, we sit in the classroom as dental students and hygiene students or health professional students, and we take the developmental courses, but we don't later translate it into actually clinical implications. So the fact that baby teeth actually begin to develop and calcify sort of in that first trimester of life hits home that, building blocks for good dental health are actually established early on. And when we think about the dental enamel, so that outer portion of your tooth surface, that is almost entirely all formed by the time the tooth actually erupts. So we know that if there are disturbances during that time frame, that can lead to altered enamel formation, which can increase caries risk if there's missing enamel or there's thinner areas of enamel. The caries process occurs much more rapidly. And I think the other thing that's starting to become more apparent is sort of an under-recognized or underappreciated risk factor are these developmental defects of enamel. So often it's the in utero period where issues of starvation or dietary stress for moms, or even with the fact that now children born premature and of low birth weight are actually, when we look at their mouths later on, we start seeing more of these defects, which increases caries risk. So I think for me, a real sort of good strong point here is, good prenatal care and diet are very important, in my opinion, for good dental development. So you really want those essential building blocks. And I guess even sort of tying on Megan's sort of talk, even the preconception period, I think, is very important too. So this is sort of the disease that I look at, so a spectrum of problems here, and sort of getting into too many pieces here of details on the slide. I think the piece that I'd like to stress home is early caries experiences basically sets that foundation for the lifetime of the child. And so we know that the kids who get caries early on are also those kids who continue to develop caries and dental health problems during childhood and adolescence. And there are negative consequences on health and well-being. And the piece that I added, sort of going through these slides here, which I have never put on this slide before when I've used it for other talks, is origins of ECC. I'm actually quite confident and strong to say that some of these origins actually begin prenatally and during infancy. And even when Megan Jones was talking about smoke exposure for young kids, we also know, and no one knows the mechanism why, but kids who are born in households where there's smoking during pregnancy and early infancy have higher caries risk. So we don't know if there's a biological mechanism or not. So just to briefly share with you a little bit here of trying to show you some of the information we have from our lab over the years, and looking at correlating mom's vitamin D status during pregnancy and the number of decayed teeth that babies have at about 16 months of age, and a direct correlation. So we do see that moms who have low vitamin D levels during pregnancy, their babies are more likely to have higher numbers of teeth affected by decay. And so I think, again, gives us sort of that example of importance of good prenatal nutrition and diet. And unfortunately, many of our women, our pregnant moms, are deficient in vitamin D given our location, and especially not just season, but if they're socioeconomically disadvantaged, they can't maintain good diets, and milk is expensive. I think here again from the same study, just to sort of highlight, when we controlled for many of these factors going on during the prenatal period and early life period, we found that the presence of these enamel defects definitely increased risk. So if kids had missing enamel on their teeth, or sort of thinner or pitted enamel, those children were more likely to have decay, mom's vitamin D level, and naturally age. So over time we find in our study, the older a child is, the more likely that they've developed caries over time. And people will often ask me, what is the prevalence in Manitoba of tooth decay in preschool kids? And we usually say about 4 to 10 kids will experience decay during that period. We can also use administrative healthcare data as well to look at early origins of caries or the consequences of severe caries. And so this was a great project, working with the Manitoba Centre of Health Policy to look at what are the early life predictors of undergoing general anesthesia to treat severe tooth decay in the operating room. And so we found that, large gestational age mattered, it increased risk. Younger maternal age, so we know moms who often, maybe they don't have great parenting supports or good knowledge about looking after young babies. The socioeconomic factors definitely matter, and income, but also frequency of prenatal care and doctor's visits matters as well. And then other things, are associated with lower risk, including initiating breastfeeding in the hospital. So I think, unfortunately, many of my colleagues over the years have really sort of given breastfeeding a bad sort of rap in the dental circles, because we know it is very beneficial. And then older mothers and children living in urban areas tend to have lower risk. So what have I learned about DOHaD along the way? I think I'm finally convincing myself that oral health and dental caries research fits into DOHaD. So Matt and I have had some conversations about that. I think there are multiple risk factors that influence a child's oral health, and including, some of these things we've never considered. So the more we talk with others in the field, the more we start realizing that there are multiple shared risk factors for different outcomes. And the other piece that sort of reinforced in my mind looking through the literature is our biggest predictor for kids for their future caries risk is past or current caries experience. And I think here the need to shift attention. So my dental colleagues, we're fixated on treating caries, so people think of the outcome rather than moving prenatally. And I think the fact as well that seeing dentistry is not part of universal health care. Or often when we talk about dental care, it's always treatment of caries. So nobody wants to, in the healthcare system, have those discussions about those investments early on, prenatally, that will actually maybe result in dividends later on for improved dental health, because they're going to say, show me the evidence, and it's tough to really prove that at times. And so I think we're at a stage where we want to be doing timely risk assessment and considering developmental origins to address a disease like dental caries. So this is some of the work that we've done for the Public Health Agency of Canada, the Office of the Chief Dental Officer, sort of tasked us to sort of look at the literature of prospective studies, looking at what factors are associated with kids developing caries over time. So we definitely know socioeconomics and special health care needs often increase. But there's some other interesting ones that sort of do focus on that early period, and I think part of that are infant feeding practices. So we want to make sure that kids are breastfed, that there's sort of, limited bottle use if possible. We want to look at these animal defects that are present and see early on, and unfortunately kids often have historically presented to the dentist once there's problems, but thankfully over the years we're getting kids in far earlier. Our recommendation is before 12 months of age, so we're starting to see these risk factors in the dentition before caries actually establishes. And then as well, there's some microbiome issues here that we need to consider as well, and as well as salivary flow. So opportunities for future DOHaD work in Manitoba. I think for me, it's sort of focusing on some of these understudied risk factors for early childhood caries, and I think for me, that's looking at the oral microbiome and microbiome as well, so I'll share a couple of slides in a moment. And then looking as well to try to document these developmental defects of enamel and see whether or not certain risk factors predispose them from occurring as well. I think we need to shift our thinking for dental prevention to be much more targeted at an earlier stage, so prenatally. That is going to be very important, especially maybe even as well early infancy, rather than only just focusing on once kids have caries and they present to us in a clinical setting. And then lastly, I think we want to be interdisciplinary and partner with others. So I think this is a great format to do that, because I now see the other DOHaD work being done, and maybe time for conversations or coffee chats just to see, are there outcomes that we should be having or shared disease outcomes that could be studied together with joint cohorts. And I'm working with cohorts as well, appreciate their benefit. So I'll talk about lastly just a couple of quick slides on transmission of bacteria. So kids, early on, they will develop bacteria in the oral cavity. I'm not quite sure what's happened there to my slide. Oh, okay. So those were those warnings. Okay. Okay. Okay, so I'll talk about, so kids can basically get bacteria from two sources. So there's vertical transmission, and it's typically from mom to child. So obviously, we also want to make sure that moms maintain a good dental care during pregnancy, because then they've got healthier bacteria in the mouths, and children will be sort of colonized by something. So I'd rather be the healthy bacteria in the mouth rather than harmful cavity-causing bacteria. The other source, though, are the horizontal transmission of bacteria. So that could be siblings, other members of the family, the toys in our offices when kids are in the waiting area, or you know, the kids at daycare. So there are those issues at play. And we want to sort of think about, because kids get colonized at an early age, can we start looking at what bacteria they have, and will that sort of predict risk? So we'll see. I've got a nice slide with nice pie wheels right now of different colors, but we have already looked in First Nations children in Manitoba to look at, are there differences between kids with severe caries and those cavity-free, and we are seeing some differences there. So that's letting us sort of think, you know, are there interactions between bacteria and diet and special populations that increase risk? So that's an important aspect. And then lastly, just to talk about, we're now embarking on research now. Dr. Chelakani from Oral Biology and I are looking at research, looking at taste genetics, so taste preferences in children at an early age as well. So when we were taught in dental school, taste buds matter, well, really not. It's really more taste genetics that are at play, and also the microbiome and how do they interact. So it's ambitious. We're looking for 800 children, 400 cavity-free kids, and the 400 kids with cavities, we will not have problems finding those ones, unfortunately. But I think this gives us a rich repository as well, where we're including not just those aspects, but good questionnaires, validated instruments, so we can have good data sets for residents and grad students in future to start sort of looking at these opportunities of what are true risk factors for caries. And these are just some of the preliminary issues that we're looking at, where we can actually look at different microbiome and now microbiome differences between the groups, and there are even sex-based differences that we're starting to see as well. So with that, I'll just say thank you, and I'll look forward to Liz Sellers coming up to the mic in her talk too.
I'll be following your studies on taste preferences. We talked about that this morning. I think that's a very understudied area, so you'll be leading the way. Our last speaker of the series and this afternoon's panel is Elizabeth Sellers. So she is in the Department of Pediatrics and Child Health here at the University of Manitoba. And gosh, you do a lot of really important work from epidemiology through to understanding pathophysiology and treatment and interventions in an effort to support youth with type 2 diabetes, particularly in Indigenous populations. So I'm very interested to hear about your perspectives on DOHaD in a slightly older cohort of individuals, because we've talked a lot about later in the lifespan and very early in the lifespan, and now we'll get your perspective.
We'll talk about early in the lifespan too. Thanks very much. So I'm going to try to keep my 3 questions in mind. So, but I'm going to focus around the next generation cohort study, because that to me is a lesson in DOHaD and really is what DOHaD means to me. I do want to acknowledge the traditional lands that we are meeting on and pay my respects to elders past, present, and future. This is what DOHaD means to me. This is a family I met in the first 24 months of my clinical career, about 15 or 16 years ago. On the day the young man who's giving us the peace sign was diagnosed with youth onset type 2 diabetes. His mom is in the picture, MJ, and I use these pictures with the family's permission, and actually they want their pictures used to tell this story. So on the day that the young man at the back was diagnosed with type 2 diabetes, when we took this picture, his older brother, he's about 2 years older, Am in the back was 9 years of age, and his brother was a little shy of 11 years of age, and they had 2 toddler sisters who were in this picture, and there's a 3rd sister who was in utero during this picture. And what struck me when I first met this young man is not only is he 9 years of age and has type 2 diabetes, which in 2002 was still a very uncommon thing to see, but that his mother developed type 2 diabetes at the age of 12 and was one of the first young ladies followed in Manitoba and recognized to have youth onset type 2 diabetes in the mid to late 80s. So within 15 years of her diagnosis, we were now seeing one of her older children diagnosed with type 2 diabetes. She was 12, he was 9. And that really was the start of my passion for this topic. So what DOHaD means to me is this family and several other families we met around the same time with very similar stories. So essentially we were starting to meet the next generation, the offspring of the first young ladies diagnosed with type 2 diabetes in their youth. We were now seeing their kids at a younger age than they were presenting with type 2 diabetes. So it was families like this and a few others who essentially established the next generation study. So the next gen study was established in 2003 within about a year of meeting this family or re-meeting this family, when we decided there's something going on and we really need to understand. And we started to follow the offspring of mums who we knew had type 2 diabetes as youth. So young ladies who'd been followed in our clinic, diagnosed with type 2 diabetes under the age of 18, we contacted a bunch of them and said, we'd like to follow your kids. And we'd like to follow them from at conception on forwards. I'll tell you, we got smarter in 2010 and started to follow the offspring of the dads. Do we wish we had done that from the beginning? Yes. Though I will tell you, we didn't completely miss the boat, because in the early days of type 2 diabetes in kids, 80% were young ladies. It's now about 60%. So we now follow the offspring of the dads. And in the next gen study, we actually followed, as of about 3 weeks ago, 268 offspring, so who either had a mom or a dad who was diagnosed with type 2 diabetes before the age of 18 and before the pregnancy with this child. So we have it's not a big number compared to big data or adult studies, but this is a pretty big number in my world of pediatrics. And we started out by just following them. We saw them every year. We tried to gather a little data about mom's blood sugars during pregnancy, but essentially we started out by seeing them once a year, making sure we had heights and weights. We then added blood pressures, we added waist circumferences, but we started seeing them once a year and measuring these kids. And at age 7, that was when we started checking blood sugars and a few other metabolic blood tests. And we chose age 7 because in Manitoba, we have a local guideline that for a child who's at risk for developing diabetes, that would be when you would clinically start screening them. So that's what we started out. And here was, and watching to see if they developed diabetes. So this is a fairly recent slide. The N is from just a couple weeks ago. So you can see, looking at this slide, we have a good number of kids, but many of them are 6 or under. So there's still a pretty young cohort. And our oldest in this next gen study is 29. So a very young cohort, and we just have a few out at that further end. If you take all of these 268 young people, age 30 or less, and most of them are under 6, 14% already have type 2 diabetes. Remember, type 2 diabetes in Canada in the adult population is 5 to 6% in the general adult population. If you look at just those who are 10 or more than a third have type 2 diabetes. And if you look at the 15-year-olds and up, almost 60% have developed type 2 diabetes. That has never been described in the world before. Our colleagues who've worked with the Pima Indian population of southwestern United States has done some similar tracking of children and systematic screening, but all of their cohorts are no diabetes exposure or diabetes exposure in pregnancy. It's a mixture of gestational and pre-gestational. So this is a unique cohort where the children have been exposed to pre-gestational diabetes. So those were pretty high numbers. Around the same time, while this data is emerging, we've shown here, but there's been a number of other groups that have shown other risk factors for developing youth onset type 2 diabetes. Clearly, pre-gestational diabetes. We have looked, as have a number of other groups, that gestational diabetes also increases the risk, though not nearly as greatly. Smoking in pregnancy, I've heard everybody I've heard today talk about smoking. We know that babies who are exposed to cigarette smoke in utero, and we know nothing about pot yet, and in the early neonatal period, the early childhood period, are at higher risk for obesity. They're at higher risk for insulin resistance by the age of 10, which is, we know, is a risk factor for type 2 diabetes. We know from observational studies that maternal overweight and obesity and rapidity of weight gain during pregnancy is also a risk factor for childhood overweight and childhood insulin resistance, which are risk factors for type 2 diabetes. So we know that quality of maternal diet in mostly actually in rat studies, but rat dams fed high-fat diets, those babies are born with fewer beta cells. So you're starting at a lower threshold. So we know quality of maternal diet is probably a problem. And we also know that food restrictions, so moms who are faced with famine, those offspring, so who have calorie limits, those offspring, again, are at risk for higher rates of overweight and obesity and insulin resistance. So that's been quite well described in Manitoba, but elsewhere, but those are all observational studies. In terms of mechanistic studies, there's very little. So we started looking at this and said, we actually need to do a little more. We can't just follow the kids and keep measuring. So the next step in our next generation study has been to look much more closely at early life exposures. And when I say early life, I'm looking at pre-pregnancy. So looking at mom's nutritional status before she becomes pregnant and dad's nutritional status before he has a gestation brewing in someone else. Looking at weight issues, looking at glycemic control issues, and also looking at those during pregnancy and in the early postnatal period. So we're trying very hard to get some of this data, including some glycemic data, looking at free fatty acids, looking at stress pre and during pregnancy. Because there's other studies that have shown that that may affect offspring's health. Our next gen study now starts again prenatally, but we also are involved with cord blood studies, so we see the babies at delivery and use cord blood to look at some epigenetics, looking at inflammation as markers of stress and some of the adipocyte hormones. So while much of this has been described observationally, we're hoping to actually be able to take it back and look at mechanisms. And why would we want to look at mechanisms? Well, that's really because then you can have some interventions. And I want to share one data on something that we have learned. So we've talked a lot about smoking today, and we've talked a lot about breastfeeding. And I'm going to talk about breastfeeding too. So this is actually administrative data. This is not. We had data, case control data from the Next Gen study that suggested that breast milk exposure decreased your risks of developing diabetes as a child. This is not from that, but it's from the Manitoba repository. So it's our administrative data. I just want you to look at the two top lines, okay? So so this is years. This is birth, and it goes to 30 years of life. And this is cumulative incidence or percent of developing type 2 diabetes. The highest line, that green line, is an offspring who was exposed to a mom with pre-gestational type 2 diabetes, who was not breastfed. And when I say breastfed here, we're only talking about initiation, which means in hospital, someone tried to help them initiate breastfeeding and they checked the box on the discharge. That could be, we do not know whether they breastfed a second past that, but in that first 48 hours, breastfeeding was initiated. So the green line are children who had a mom who had type 2 diabetes before the pregnancy, who were not breastfed. And if you look at 25 years, about 12.5% of those offspring have type 2 diabetes by 25 years of age. By the shake of a dice, half of those are going to be girls, and they may not have had their babies. So now we're exposing another generation. If you look at this solid blue line, the dark blue line, these are offspring, so children as they grow up who were exposed to a mother who had type 2 diabetes in her pregnancy, but she breastfed, meaning she initiated breastfeeding in hospital. That is a massive change. That's a halving of the risk for developing type 2 diabetes by the age of 30. There's very little in the world of medicine. Listen, if I developed a drug that decreased your cardiovascular outcome risk by 5%, like I would be world famous and I'd be very rich and very retired. This outstrips that. And I'm not going to sell it to this audience, but there's many other health benefits of breastfeeding to both mother and child. And we have been involved in some of those studies looking at breastfeeding protecting mom who has risks for type 2 diabetes, preventing her onset. So this is the good news story, which is why we're kind of turning our attention to the developmental origins of health. How do we help keep this population health or improve their health? And I think the opportunities in Manitoba are fantastic. They are wide. So my life view is very much the life course. Alex is gone, so he could hammer me, and I don't get a chance to hammer him. Pediatricians do look beyond 18 years of age. We're not as set up to it, but it's really a life course. We have the opportunity in Manitoba to be looking at pregnancy and pre-pregnancy issues in the mom, including things like, well, I'll give you an example. We have the opportunity to look at delivery mode, at the microbiome, at the location of delivery. Let's think outside the scientific world. Does it make a difference when you take a mom from northern Manitoba and you take her out of her home for 4 weeks and she delivers in Winnipeg with no supports? First of all, that probably decreases breastfeeding, because you need your supports. What does that do to maternal stress, cortisol levels in that last 4 weeks of pregnancy? What does that do to the baby? We don't know. So we have the opportunity to look at that. We certainly have the early years with many different into adolescence, into young adulthood, and then the next generation. And I can't forget the dads, because they play a role here. So in Manitoba, with type 2 diabetes unfortunately being the data is not actually published yet, but we will have the highest reported incidence anywhere in the world, and it's quite a bit higher than in the southwestern United States. We have the opportunity to make a difference. What I think we need to do when we look at the life course is to look at the life course with all of the expertise we bring. So I'm a clinician and a clinical scientist. We've got basic scientists. We also have people in social sciences, and we have community members and communities. And I think we need to look at it at all those levels. So we need to look at it at the individual and family, the community level, and a systems level. So I'm going to give you a quick example, and then I will be quiet. So if we think that maternal stress is important and maternal nutrition is important, so absolutely, we can use some of our animal studies to look at quality of maternal diets and effects on offspring. But we also need to look at safe drinking water, housing, food security. That is not an area to which I bring expertise, but I have colleagues in the community, in public health, in leadership, who can help with those things. And that, of course, is some of the issues of inequitable access to health care, where you deliver, may make a difference. So again, we need the policymakers, the community leaders involved in here. And of course, that will affect food access, safe environments for infants, young children as they grow up, and the cycle continues. So I think we need to look at our own areas of expertise, but we need to look outside the traditional academic world. I don't know if that makes sense to you. And that, oh, no, I do have one more picture. Huh, maybe I don't. I do, it's not moving. Here we go. So, I want to bring you back to our family. So this young man was 9 years of age when he was diagnosed, and his mom was 12 and a half. Let me update you. So this is the family in a more recent picture about 3 or 4 years ago. You can see that they are all grown up looking, and the eldest brother is unfortunately not in this picture. He just wasn't available. What we now see the young lady who was in utero in the first picture. M had developed type 2 diabetes in this picture. All 3 of the young ladies have developed type 2 diabetes of youth onset, and 2 of them now have children of their own. So Alan was talking about the generations. Unfortunately, we are now into 3 generations within our next generation cohort, because we have data on some of the mums, like this lady, their offspring, and some of those offspring now have offspring. So fortunately or unfortunately, we are now getting into 3rd generation. So the cycle continues, and our mantra now is the developmental origins of health, to help the offspring of the 2 babies who aren't in that picture. Unfortunately, the mum we lost last November. So the story and the learning that we're learning from this family continues, and hopefully we can change the trajectory of that if we work together. So that is my story. You told me 10 minutes.
Time for questions. If you both want to come up to the table, will you turn the table mic on, or it's on? Okay. Do we have any? Yes, burning question right off the bat.
Somatic or otherwise mutations in the genomes of these patients.
So are you talking about youth onset type 2 diabetes when you say that? So there is a polymorphism or a variant of a specific gene in some of this population, kids of OG Cree origin, which are about 30 to 40% of the kids we see. They are not all of them. It is a polymorphism or a variant because it's present in well more than 5% of the population. We have pretty good historical data to suggest that diabetes didn't occur 75 years ago in this population. If you look at medical records, station records, there's nothing to suggest that it occurred. Whether there was something advantageous to having that variant, we are interested in and looking at. And Kristin Doucette, who is an islet cell biologist at, I'm going to say Mitch, I always get it wrong, at Crim, Mitch, at our place, is looking at whether we have some suggestion in an animal model, because we've developed an animal model with that variant, that they store fat differently. So clearly in a time where food stuff was available, not available, available, not available, the ability to store fat was advantageous. So that's a piece of it, but it is not the whole story, because we now see kids from, I think, 60 of 63 communities, First Nation communities. And when we say First Nation, I think it's important that, because it is 87% of the youth in Manitoba with type 2 diabetes are self-declared First Nation, but we need to remember that that's kind of like, it's almost a bit like saying I'm pasty white, because there's quite a big difference linguistically, traditions, history, genetics from a Mi'kmaq on the East Coast, OG Cree here, and on the West Coast. So even within Manitoba, there's probably, we need to be careful we don't paint with one brush. So it's a piece of the study, but not all of it.
But for this particular family, do you have any genome?
Well, yes and no. We have limited. We have not done a genome-wide scan, for example. We're actually really interested, as is the family, in the eldest brother, because he has not developed type 2 diabetes. So he's actually really interesting. So he has wanted to and has participated in, we're actually haven't got it all back yet, but we're looking closely at him. N of 1, but sometimes that can teach you something.
And we talked about that this morning, reframing things and what protects individuals, allows them to be resilient, not always focusing on pathology.
Yeah. This is not the only family we have like this. So.
Other questions for panel? Yes.
I think this panel is really good. We've talked about knowledge translation and informed research and research priorities, and I think this group of panelists in particular work with underrepresented populations. And I'm just wondering, it also offers an opportunity for mixed methods research. So I'm wondering if you can speak a little bit to patient engagement, knowledge translation, how we give those groups a voice and allow that to then work back and inform our research priorities. Just thoughts.
Maybe I'll go first. I think including mixed methodology is very important, and I think over the past few studies that we've done, and maybe they may not necessarily be true DOHaD, but when we've done program evaluation for government programs targeting young kids and their families, we actually have Dr. Kayun Aiken at the back there. She's our postdoc fellow right now, so she's a qualitative researcher working with us because we see the value of asking important questions for community members of what are the best methods to improve oral health, because we can't just give a fact sheet and leave the community. And as well, even if communities are asking for certain things, again, the one-size-fits-all approach, for Indigenous communities is not appropriate. And so even working now closely with a few very geographically close communities of Pine Creek, Duck Bay, and Camperville, we're even hearing, don't give us a report for all 3 communities lumped together, even though they're basically 5 minutes apart by car, but we want distinct information that we can then sort of use at a community-specific level because we're governed differently, we have our different leadership structures, so I think that's important. And then tips of what works, what doesn't, and even with some of our preventive, secondary prevention, hearing views of, yes, that's a clinically acceptable one, but we don't like the side effects that treatment might lead to the teeth to manage caries. So I think there's great value in hearing from parents and grandparents?
So yeah, I think that's super important. We are learning. We are doing some qualitative stuff, but so anybody who's out there training in qualitative research, that's actually a hole in our own sort of program in terms of, yeah, we're doing a little, but we're missing some real expertise. And when I say our research program is really DREAM, Next Gen, we have a cohort of kids who live with type 2 diabetes called the eye care cohort. We're working really hard. It is slow and we're learning as we go. We have a patient advisory committee that's part of our eye care cohort. We've had a community advisory committee with Next Gen since its conception to help inform what are the important questions, but also then translation. So for example, the youth who sit on the eye care advisory committee have well, they've done all sorts of things we weren't looking for, but they wanted to develop a video to send their message, because from their point of view, the biggest barrier to them helping themselves in terms of self-care once you have type 2 diabetes, the biggest barrier that they identified was blame and shame. That was a bit of a surprise to us. We knew that it occurred in the medical system. We know that's a problem. But from their point of view, it occurred in their families, in their communities, in their schools, so they've also developed a working workbook for other youth with type 2 diabetes. So they kind of looked at our resources and said, yeah, no, we could do better, which is true. So it's slow steps, but certainly for the work that we're involved, the advisory committees, which include people who live with type 2 diabetes, but also stakeholders in the community, leaders in the community, has been really important.
I'll speak to this a little bit differently because my underrepresented population is really the population of the dead. There is a high mortality rate in catastrophic neonatal illness, and those patients are not going to be returning to focus groups, and quite frankly, neither will their families, because this is traumatic in the extreme. I'm going to share with you that I actually recognized the extent of that only a few years ago. I had gone back to India, and I was talking with family members about what do we do what is the research you do, and I was having a nice chat with my aunt and uncle, and then I just looked at my aunt's face, and there were tears standing in her eyes, and I realized she revealed to me then she had a son who was born around the same time I was, who died of pulmonary hypertension, and for 50 years she didn't talk about him. And she only spoke about it because we were discussing the research, and that 50 years later, yes, he might have survived. And that was so traumatic to her that she spoke at that time, and then she's not spoken about him since. And I didn't know when I started doing pulmonary hypertension research. My cousin had died of it. But I think they're underrepresented in our conception of who could be stakeholders, because these are people who do not speak of their experiences, internalize it, and would never present themselves to a conversation about it. And this, I think, is a blind spot we have with respect to dissemination of both research and also how do we talk about these things. And one of the ways that I've started exploring, and some others in medicine explore much better than I, is the idea of narrative medicine, of trying to use storytelling to give voices back to the missing voices. And that is an aspect of the qualitative, it's not really qualitative research, but it is qualitative re-representation of voices.
I think we have time for last question. Great.
Instead of a dream within a dream, because you've got the issue of the condition that you're interested in, early caries,
I'll answer quickly because I think mine is the much smaller piece of this. Environmental influences on longitudinal health are not just are not necessarily chronic exposures. They can be acute catastrophic exposures such as hypoxia. So that gets at one of the aspects of DOHaD is environmental risk. This falls into that category. The other longitudinal impact is on rehospitalization. If I talk to a pediatric intensivist, they will blame me for supplying them with 50% of their patient population. And they're probably right.
I gotta say something. Okay. Bob is actually part of our world. No, and we talk a lot because some of the same risk factors are also risk factors for the development of type 2 diabetes. And oral health is actually an important part. Periodontal health is related to cardiovascular risk in type 2 diabetes. So our risk factors are the same. We use his bottles full of sugar. I mean, as
a teaching tool, not to promote caries.
No, but Bob's group developed some teaching tools through the Healthy Smile program, which are completely relevant to our world. So we just stole them.
I think, there's a lot of recognition, or at least increasing recognition, for sort of the early period now that's growing. I think the piece, Andrew, that I think time will show, as well as sort of the long-term oral health or other health consequences of maybe kids who develop severe dental problems early on. Part of it is right now we don't have good data, and I'll sort of steal Megan's sort of unicorn idea. Like when I do have dental colleagues who sort of think about sort of these long-term consequences, because we know there have been some researchers looking at birth outcomes and periodontal disease in women, and sometimes some studies come out with yes and other studies come out with no, and so there's a lot of, I think, misinformation out there, or maybe a lot of clinicians who just don't know what the current state is or don't know how to read the literature well, so they're a little bit skeptical. But I think over time, we're going to see, even whether it be clinical data or administrative healthcare data, that will allow us to answer those questions. But I'm hoping as well that clinical colleagues over time will see the value of good clinical notes and integrating oral health records into our electronic medical records. So let's be honest, we have hospital dental clinics here and community-based dental clinics, and often situated within the same building as an EMR, but the electronic dental record doesn't interface, and in future, I sound like Alex now, sort of integrate those, but you can actually look at sort of those opportunities to study sort of things over time.
Okay, I'm going to be a little sarcastic now. Yeah, that's true, but within our own pediatric hospital,
we
have more than one database that doesn't talk.
Yeah.
That's the truth. Most people don't believe that.
Liz, I'm going to come back to you on one point. There was some data, actually, I saw literally last week, that makes a very strong link between infants of diabetics and increased risk for pulmonary hypertension in the newborns. And
that's why I didn't do neonatology. Actually, it was being on call with PPH.
Well, it may be that infants of diabetics have different hypoxia tolerance, and therefore it's a two-hit kind of phenomenon, that not only the disease is more frequent, but it's more severe.
In this province, we actually have a tool, which is our administrative data, to at least get a signal that that's worth pursuing.
No, and in fact, we don't have good animal models that will address any of that, because if you can look at the mama animal, you can't look at the baby animal, and there are a lot of gaps in the animal research that could be addressed by the population research.
Okay, I think we can give the panel a break. And everybody thank the speakers for coming this afternoon. I think Cosetta is going to have some parting words. I just want to say I loved hearing the talks today, these rapid fire talks on what DOHaD means to different people and different disciplines. And I learned a lot about the richness of the research data and the administrative data that exists here in Manitoba. And as I said this morning in my talk, I think you're very much positioned to be massive players, not only nationally but internationally in this field. And hopefully everybody can start talking together some more. We can get these data linkages happening, and you're positioned to do some great things. So thank you.
Awesome. It has been a wonderful day, and I'd really like to thank our keynote speaker, Dr. Kristin Connor, for her spectacular talk and her vision in the field of DOHaD. I'd also like to thank all our amazing guest panels, Dr. Azad, John, Singer, Dakshinamurti, Professor Shroth, and Dr. Seller for their insights and recommendations on opportunities for future DOHaD work in Manitoba. Yesterday, when we were discussing with Dr. Connor about her talk today, she said that I'm going only to scratch the surface about the field of DOHaD. And I believe after what we listened today from our keynote speaker and all the other panelists, that's true. And this masterclass is so important, especially from a training perspective, because there are several trainees that they work on the field of DOHaD, but they do not realize that. And I think this workshop helped, or this masterclass helped them understand that they are working in DOHaD, and DOHaD is so broad. And this masterclass also creates strong foundations for the developmental origins of health and disease curriculum. The DOHaD Network, together with the support of our institute, the Children's Hospital Research Institute of Manitoba, has committed to developing a curriculum which will be available to all the trainees within the institution, faculty, research technicians, support staff, stakeholders, and patient partners. I think throughout the symposium today, Dr. Connor and our local guest panel experts have amply demonstrated the interdisciplinary aspect of DOHaD research, and we can see that that interdisciplinary strength doesn't just enhance DOHaD, but it's the central to the progress of this field. And it was very telling this morning during Kristin's talk when Dr. Haleko said, help us to see where we want to be in DOHaD and how can we get in there. And she gave several examples of unanswered crucial questions and emerging fields. For example, the vulnerable populations, the transgenerational research, programming fathers. And also she went through and identified barriers to conducting DOHaD research. And these things are central part of DOHaD research, and this is something that we may want to accomplish within our institute for the coming years. And I think with the kind of support that we have from Devotion, from CREAM, the university level, and the talent of people in this room and our colleagues who are not today in this room, we have great things in front of us in DOHaD research here in Manitoba, Canada, but also worldwide. So I'd like to just close the day by thanking some people without whom this wouldn't have been possible. Dr. Haleko acknowledged some of them this morning, but this day is a culmination of the effort of a team who worked for the last couple of months and helped bring about the reality of the campus-wide interdisciplinary DOHaD research. And I'd like to thank our amazing Devotion Network, Erica, we have all seen emails from her, and she has done lots of the arrangements. Rachel, Dr. Haleko, and Dr. Jennifer Prodiger, who couldn't be here today. And they have really worked hard on this. Thank you to all for making this possible. And from my own experience in the last couple of months, there is one person who we could have never done this without. And she really kept us on focus, narrow, kept us organized, and took care for everything behind the scenes. And that's Natalie Rodriguez. Thank you. So the next DOHaD Masterclass, you may be interested in that. That will be on October 21st, and we will have a speaker from the University of Bristol, Dr. Caitlin Wade. She will talk more on the methods of DOHaD research, especially Mendelian randomization studies, randomized control trials, so it will be very interesting. Then after that, on the first week of March 2020, we will have another amazing Canadian researcher. She will talk more about the mechanistic studies, and that's Dr. Sandra Davidge. So thank you all for this fantastic symposium. You'll be hearing from us, and we will be hearing from you. Our feedback is really important to us, and as Dr. Haleko said this morning, this masterclass is recorded and it will be available in Devotion website. Keep an eye on Twitter. We will notify you there. So please join me in thanking our keynote speaker, Dr. Connor, and our local speakers, and of course to all of you that were here during all the day today. Thank you.